into life sciences. And then once I was into this entire field of life sciences, I think I then recognized, as many others have, that probably the most interesting questions, at least to me, that my generation of scientists has, is in living matter rather than in inert matter. So that's why I decided to stay. And since then, I have not stopped finding interesting questions. So that's why I'm here, I guess.
And you know, like, maybe can you comment, you know, like there are lots of like young like students who are potentially interested in mechanobiology, but they're maybe like a bit afraid of like learning all the messy biology, right? So do you have like any advice for them, like how to overcome this like fright and like this barrier?
Well, I think so. I would say life science students are more frightened about equations than physicists about the messiness, no? And I think you used the right word, right? Messiness. That's the biggest problem that physicists and engineers have in biology it's messy, right? It is so diverse, right? So there's so many thousands of genes and proteins. They can bind in so many ways, right? So how can there be any sort of order or any sort of predictability in a system like that, right? And this is what really prevents, I think, lots of physicists in joining it. But then there's these big principles, right? I mean, there's principles of robustness and reproducibility, and it means that there are laws, right? And this is what physicists do. And I don't think you need to understand so many details. And I think this is a good thing that physicists bring to the field of life sciences, which is that we don't really like details, right? And whereas life scientists often love details, which is also good. You also need people who love details, right? But our love for like these first principles, laws, universality, right? I mean, to bring these ideas to life sciences, I think it is very important, even if we know that life science is also about diversity, right? So, but I think this is completely compatible, and bringing the two schools together is actually very useful.
I totally agree with you. And can you comment, as you said, like, you know, like all these like life scientists, they're really like afraid of, you know, like equations and these general principles. So like, how do you overcome this like language barrier with them to, you know, make them appreciate the kinds of things you're doing?
I think, so my advice is for them to embrace this language, right? So this is the language that nature speaks. And therefore, I mean, you don't need to be an expert in solving complex differential equations, but you need to understand this language. So otherwise, you're very limited. And I have examples of life science students, PhD students in my lab who have come, and there's no reason for them not to learn it. No? So sometimes it's a barrier that they built to themselves, or maybe during their undergrad, it's a barrier that even faculty gives to the students. No? But then once they recognize the importance, and maybe that's the most important thing, is to convey that this is really important, that it's very hard to do science without equations. And that once you embrace the equations, this is very powerful. And so I think this is the message. So so I wouldn't say it's okay, you don't like equations, you don't like computers, well, you'll be fine. Well, that's not what I tell my first year PhD students. You know, what I tell them is embrace math. It is not that hard, at least at the level that you will need it, but just do it. Just do it. Because once you do it, it's really extremely powerful.
Great. So we have like a number of panelists here already, like a few more will join. So, you know, like maybe we can just go briefly around and we can introduce them. So, you know, like maybe just I'll go around the screen and just briefly introduce yourself, like, you know, what's your name, like what's your affiliation, and maybe also like tell us like something about your interests, like research interests, and then we can, you know, like also open the floor to panelists to ask other questions to Xavi, right? So maybe we can start with Matthias Merkel.
Thanks, Andrej. Hi, everybody. So my name is Matthias Merkel.
You're very soft. Can you speak louder?
So yes. So let me just bring up my microphone. Okay, let me do this. Sorry. Okay. Do you hear me better?
Yeah.
Yeah. Okay. So hi, everybody. My name is Matthias Merkel. I'm a junior group leader in France, in Marseille. And so here I'm working at the Turing Center for Living Systems. This is a new interdisciplinary hub that brings together engineers, physicists, mathematicians, biologists. And so, yeah, and I'm excited about collective cellular behavior. On the one hand, I'm interested in close collaborations with biologists, and there I have a lot of opportunity here. And on the other hand, also thinking about hard physics questions.
Thank you, Matthias. Rastko?
Yes, hi everybody. I'm Rastko Snepnek. I'm a senior lecturer at the University of Dundee in United Kingdom. So senior lecturer will be roughly equivalent to the associate professor to the rest of the world. So I'm by training a theoretical physicist I've been trained as a kind of low temperature quantum kind of superconductivity, quantum phase transition person who kind of gradually transitioned to soft matter, active matter, and eventually to biophysics. My research is focused in the School of Life Sciences at University of Dundee, which is a very large center for biological research, mainly for what's happening at the microscopy, at the molecular scale. But I'm closely collaborating with Xavier, who is a developmental biologist. So we are looking into trying to understand the mechanochemical coupling in early stage chick embryos, essentially how the chemical and mechanical patterning coordinate together over the millimeter scales to actually form the flow patterns kind of morphogenic flow patterns in early embryonic development. Thank you.
All right, then we have Daniel Matos Fernandez.
Hi, everyone. My name is Daniel Matos. I'm an assistant professor at the University of Warsaw. And I work mainly in active matter, and I like to focus on chemical feedback loops. So I'm working mainly in trying to understand basically the out of equilibrium aspects between competitions between chemical interactions and mechanical forces at the mesoscale level. And I'm just working before as a microbiologist, as a physicist, so doing experiments. And in there is where I just get to love everything about biology and just learn a lot about it. A little bit what Xavi was saying, not being afraid of asking questions and just listening and paying attention to more details than as a physicist would like to. Yeah, so that's all.
Thank you. Then we have the other Daniel Sussman.
Yeah, hi. I'm Daniel Sussman. I'm an assistant professor at Emory University in the physics department. My training is in soft and active matter, and I'm really interested in thinking about kind of minimal models for emergent and collective behavior of living systems, both at like the dense cellular scale, but also the full organism or organismal scale.
Thank you. Then we have Jianping.
Oh, yes. So my name is Jianping Fu. I'm currently a professor in the mechanical engineering department at the University of Michigan, Ann Arbor. So my research these days, we are using human pluripotent stem cells, including embryonic stem cells, to develop models of early human development, including both human peri-implantation human development, as well as human neural system development. So we are studying this developmental events using human pluripotent stem cells. We are also very interested in understanding how mechanobiology play a role in shaping the tissue and organ formation, especially how mechanobiology directly act upstream to mediate downstream developmental signaling events. Right. Thank you.
Thank you. Sohan.
Hi, everyone. My name is Sohan. I'm an assistant professor in the mechanical engineering department at Virginia Tech. And I'm coming from mechanics background. My training has been in homogenization of composites and those topics. So I'm coming with a materials perspective to understand how living materials behave and how mechanobiology, from the modeling perspective, how we can understand various feedback loops in active processes, including cell migration, collective cell migration, and mechanics of collection of cells.
Thank you. And then Satoru Okuda is joining us from Japan.
Hello. I'm Satoru Okuda, associate professor in Kanazawa University, Japan. I'm interested in mechanics in self-organizing muscle dynamics in 3D space, especially the cell resolution understanding of the muscle dynamics. And now, in these days, we are focusing on the interactions between cellular and tissue dynamics interactions. And we are trying to develop a new methodology, like a computational methodology of 3D Vertex Model and other new computational methods. Yeah, thank you.
Thank you, Satoru. Matej.
Hi, my name is Matej. I'm from Jožef Stefan Institute in Ljubljana. Mostly I'm developing theoretical models of tissue mechanics at different scales. My most recent interests are in active dynamics at the cell junction scale, but I've done also some work on mechanics of tissue folding and wrinkling, and also on tumor growth and how tissues interact with supporting structures such as basement membrane.
All right, and then I have a few other people I see here. So we have Ying Zhong.
Yes, hi everyone. My name is Sarah Ying Zhong. I am an assistant professor at the University of South Florida. So the major focus of my research is in the field of advanced manufacturing. So one direction is a portable and ultra-fast and repeatable disinfection technology for surfaces and the environment and PPEs. And the other direction is ultra-fast printing technologies for flexible sensors. And currently we are stepping our foots into the field of 4D printing in controlling the differentiation of stem cells. So I think this seminar will be a very great help for our research. Thank you very much.
Thank you, Ying. Then we also have Teng Shuan.
Hello, everyone. My name is Teng Zhang. I'm assistant professor in the mechanical and aerospace engineering at Syracuse University. My research is focused on the interface and the instability of a material, both in biological and engineering material. One focus now is the shape morphing of soft and active material. And we show this shape morphing in pasta, the food material. And we are also very interested to see how the shape morphing in the tissue and also how to use engineering the hydrogel elastomer shape morphing to guide the tissue like regeneration. So this seminar will be very helpful for my research. Thank you.
Thank you, Teng. So we'll have a lot more panelists joining us later. But now maybe we can also introduce to everybody the core team of the EML webinars so that like also our panelists get to know them. So maybe let's start with Teng.
Hello, everyone. My name is Tang Li. I'm a professor in the Department of Mechanical Engineering at the University of Maryland. My group studies the mechanics of materials, including the nature-based sustainable materials, low-dimensional materials, energy materials, and in recent years, we also start to look into the machine learning-enabled material discovery. I'm also an editor of EML.
Thank you. Jimmy?
Jimmy Xia here. I'm a professor at Nanyang Technological University in the School of Mechanical and Aerospace Engineering. My research interest is mainly mechanics. Recently, we've been doing something related to cell mechanics, mechanobiology, but also plant mechanics or mechanics of plants. I'm also editor-in-chief of EML, together with Zhigang and with the young people like Tang Li and Sulin Zhang.
Thank you, Jimmy. Sulin?
Sulin Zhang from Penn State. So I'm one of the editors of EML. So my research interest is on the mechanics of living, mechanics of active materials, both living and non-living active materials. For living materials, we are mostly interested in mechanobiology. And so I'm very interested in today's talk. I'm looking forward to it.
Thank you. Zhigang?
Zhigang Shuo from Harvard University. My field is mechanics of soft materials. I also live in messy world, engineering, very messy. I embrace messiness. Thank you very much.
Thank you, Zhigang. So I see a few more panelists showed up. So we have Jeremy Green.
Hi, Jeremy Green. I'm based at King's College London. And I'm a sort of card-carrying developmental biologist interested in morphogens and morphogenesis, looking at both epithelia and mesenchyme and using lots of sort of live imaging techniques and a bit of modeling where I can.
Thank you.
So I like working with messy engineers.
Thank you, Jeremy. Then we have a Liqing. Yeah.
So I'm working in Paris in Institute of Global Physics in Paris. I'm working on lithosphere mechanics. Yeah, I have already followed this webinar for more than 1 year already. Yeah, thank you.
Okay, we have Hai Chao Han.
Oh, hi, good morning. Yeah, this is Hai Chao Han at University of Texas, San Antonio. My interest in hard blood vessel mechanics, look at the tissue remodeling in the heart and in the blood vessels, and also like the mechanical instability, buckling phenomena. Thank you.
Thank you. And then we have Taher Said.
Hi, good morning. At least from my side, I'm in the University of Illinois, Urbana-Champaign. Hi, Xavier. Good to see you again. My interest is in collective behavior of living cells. They act together by communicating with each other mechanically and emerging into living machines. So mechanobiology combined with some extreme mechanical thinking.
Thank you. So I think we've gone through the whole screen. So we have quite a diverse panel, like there will be more joining up later. And we're really looking forward to the extended discussion after the Xavier's talk. So we already talked like earlier when some of you haven't been here yet, we talked a little bit about Xavier's very interdisciplinary background and how to bridge the two worlds, like of physics, engineering, and biology. And now I'm happy to also give opportunity to other panelists if they want to informally ask some questions to Xavi. Yeah, go ahead, Rastko.
Yeah, so hi, Xavi. So I was kind of, you said some very interesting thing about the that the biologists have to embrace the mathematics, which I completely agree with you, right? But how do we go over this, in your opinion, about this cultural barrier? Because very often when you talk to people who went into kind of biology route, they say like, well, I was not very good in mathematics in school, to put it very kind of bluntly, right? And now you want me again to go back to doing mathematics. So how do we bridge that gap? So what do we tell them?
Well, I would say that there are probably some impossible cases but in general, I would say that I think the key is to make them see that maths are useful for their projects, right? And this is now, for example, there's so much microscopy, for example, right? So you're so much more powerful if you can program a computer that does basic matrix operations. If you can do that, if you're a biologist that knows this compared to a biologist that doesn't know how to do this, the difference is so huge. that I think all these students, they will see it, they will find it useful, and many of them will also then find it fun, because at the end, many of us here who are on the sort of computational side or more physical side, I think we have fun when we write models, when we solve equations with computers or by hand, right? So I think at some point they will see that it is fun, but I think the key is probably usefulness, right? So these days, being a cell biologist or developmental biologist, without knowledge of computer science or some basic math, it is very hard. You're not competitive in the market, right? So I think that at some point, I mean, this is the language that nature speaks, no? So you have to speak it.
Thanks. Daniel?
I guess that I have a follow-up question on that. It's about how you actually pick up the systems, right, in biology, because they are so diverse that, let's say that you wanted to prove a point, right, or, you know, and then you have such a diverse systems and you have such a diverse behaviors, right? So as a physicist, when trying to generalize, right, whereas biologists, they're trying to be more specifics about, you know, let's say this is particular, you know, type of, you know, cell, it does that particular thing, right? So my question is, actually, how do you, when you say, okay, I wanted to just understand forces, let's say on, you know, substrates, and you say, well, why you pick up the system that you picked up, and, you know, when there is so many systems out there, right, and in a way that you can just generalize it.
Right, I guess this is the hardest thing to do in science, to pick up the system and to pick up the question. No? So how, there are so many interesting questions, which one do you pick up? No? And I think this is a question that I would like to ask to a lot of people, right? Including many of the panelists here, right? In my case, I try to identify systems in which we can have an impact, in which we can change the way we think about it, in which we can really bring something new. And this is not always the case. So we try to find systems that are simple enough so that we can understand them with some additional measurements. But at the same, they are sort of complex enough so that they have some sort of emergence to them, right? So that's something that we always find in systems. We try to find emergence, but we also try to find simplicity, right? So, so complex but not too complex, simple but not too simple, right? So this is where we try to stay. And this is, in fact, in my talk, I would talk about different systems. And maybe I can sort of answer to this question as I go on, right? Because you will see that we're now going into systems that have incremental complexity, but that one thing leads us to another. And now we are working with organoids, for example, in which you have different cell types, stem cells, differentiation, all sorts of problems. But we're at a point where we can actually make an impact and really understand how this works. You know, so that's at the end, really picking up a system in which you can give strong answers, right? You can say, I think this is how this works. This is something that is really important for me, right? And this is something that in living system is not so easy, right? Because they are so complex that really finding interpretations that where you can really be conclusive, it's actually very hard.
Great. So Jimmy, we're approaching like the hour. So like, I know that usually you share like a few slides before, like, is that the time or should we keep going?
Yes. I was going to say that. Thank you very much. First of all, while I'm sharing my screen, I'd like to again thank Xavier and Andrej for, first of all, giving this webinar. We're all looking forward to it. And also for putting together such an impressive panel. And it's just marvelous. Do you see the screen now?
If you can change to the presentation mode, the second one, drop down list.
Okay.
The display setting.
Display.
Above the slides, there are 3 options. And the middle one, there are a drop down list.
Hold on. I was able to do it last time.
In the top left corner near that. Yeah, the middle one.
I don't see.
The display setting, that's a drop down. Yeah.
Is this okay now?
Yeah.
Good.
Good.
Yeah, very good. Very good. Okay. Welcome to the EML webinar. And EML is organized by this journal, Extreme Mechanics Letters. And Extreme Mechanics Letters, as you can see, seeks to publish research of immediacy, depth, and originality. And we have team in EML, together with John Rogers and Zhigang Suo, three of us serve as editors-in-chief. And we also have a very dynamic and active group of editors, Katya Taldi at Harvard, Hanqi Jiang now at Westlake University, Teng Li at University of Maryland, Madin City at Max Planck, and Suyin Zhang at Penn State. Our publisher is Rebecca Capone at Elsevier. Just want to share a few recent publications to give you a flavor of what we do or what Extreme Mechanics Letters publishes. We had a recent perspective written by Madin City on physical intelligence as a new paradigm. Fascinating perspective, talking about these intelligent systems without the brain. And back in 2020, we also had publication by Yung Zhang and co-workers at Delft's University of Technology on design and characterization of multi-stable mechanical matter structures, level and tilted stable configurations. There's a little picture on the left to demonstrate what they mean by that. And also recently, there's a paper published on the shear wave elastography method for solid tumor, especially for the effect of interstitial fluid pressure on this particular SWE method or shear wave elastography method. It's a method commonly used to distinguish cancerous tumor versus benign tumor. So this is a paper addressing the mechanical part of this. EML Webinar started last year. Zhigang, you want to make a few comments on this?
Yeah, thank you. Yeah, EML Webinar started as in the early days of the pandemic. So we all felt by now that a pandemic really should leave. EML Webinar should stay. So EML Webinar has been doing quite well. Thank you.
Okay, and you see the statistics over there. And this is season 2. This year we started season 2, and we introduced a few new things. First of all, we emphasize interdisciplinarity, and we also publicize it on a number of different platforms. And we also introduced a new live streaming by WeChat. And for example, one of the EML webinars on the WeChat live streaming, we had more than 12,000 participants or viewers. These are the webinars that we've had so far in season 2. And Xavier is the latest one today. And we also started something new, or we started an experiment. Teng, you want to mention this?
Yeah, thank you, Jimmy. I would like to take this opportunity to thank all the supporters of EML webinar series, all our audience. As you see, we see many people have been attending EML webinar every time. And in season 2, we start to run EML webinar independently. We acknowledge the technical support from Harvard School of Engineering Science in season 1, and in season 2, we started fundraising event to help raise the minimum level of funding to support the running of EML webinar. And we have been very successfully completing this fundraising event, and we acknowledge all the donors listed in this slide, and we look forward to your steady support in all the webinars upcoming in the future.
Okay, thank you very much, Tom. So if you want, you can still donate to this GoFundMe site. So let me stop here and turn it back to Andrej to introduce.
Thank you. Thank you. Xavi, do you want to start sharing your screen while I introduce you?
Yeah.
All right. So welcome everybody to today's EML webinar that will be given by Xavier Trepat. We're really delighted to have him. So Xavi is currently at the Institute for Bioengineering of Catalonia. So Xavi's background is very interdisciplinary. He got degrees both in physics and engineering, and then later in medicine. And Xavi very creatively combines all of these interdisciplinary approaches, and he has brought a lot of innovation into the field of mechanobiology. And he has been also extremely successful. For those of you who know European Research Council grants, ERC grants, Xavi has pretty much won every single grant that's possible. He has won the starting grant, consolidator grant, advanced grant, and even the proof of concept grant. So we are really delighted to have Xavi here, and we're looking forward to learn more about your latest development in engineering the shape and mechanics of cellular monolayers.
All right. Can you see my slides?
We do.
Okay. Thank you very much, Andrej, for the very kind introduction, and to Jimmy, Jigang, Zulin, thank for putting together these webinars. I think it's a very original series. These long discussions are very insightful, and I hope that when the pandemic completely goes away, hopefully soon, this will continue, because I find it really interesting. Okay, so I'll tell you today about how we think about epithelial monolayers, and like the one that you see in this picture. One of my favorite epithelia. This is the intestinal epithelium in a mouse, and I will be telling you about this system later in my talk. So essentially, every internal or external surface of your body is covered by these thin layers of cells that are called epithelia. They come in many different flavors. Sometimes they are just one cell thick, like in this case. Sometimes they are stratified. They are tightly packed. They are connected to each other through different protein complexes, like tight junctions, adherence junctions, desmosomes. And they are usually, but not always, adhered also to a soft hydrogel that is called the extracellular matrix. Now, despite the fact that they account for a very small fraction of the mass of your body, they play a fundamental role in a number of physiological and pathological processes. And for example, they enable tissue compartmentalization. They are the first barrier in the protection against pathogens and against physicochemical agents. They drive morphogenetic movement, especially in the early embryo. They transport ions, fluids, macromolecules. They enable wound healing, and they enable tissue regeneration. And unfortunately, they are also the origin of about 80% of all tumors. Now, in order to perform all these functions, epithelia must act as what I call mechanical multitaskers. Epithelial cells must be able to migrate, to divide, to adhere, to fold, to differentiate, to die, and so on and so forth. So all these functions are mechanical. You cannot move without a force. You cannot divide without a force. Same thing for adhesion, for folding, and so on and so forth. And I call them multitaskers because epithelial cells often perform many of those functions at the same time. So the question that I would like to serve as the umbrella of this entire presentation is really how mechanics drives and coordinates these tasks in time and space. And I will begin telling you a bit about wound healing and about cell migration. So this is a movie that we recorded in the lab about 10 years ago now. And but we could have recorded at the beginning, this movie at the beginning of the 20th century, because essentially all you have to do is take one of these layers of cells and you scratch them either with an object or with a laser, as we did here, and then you just watch how cells scroll into the wounded area. So some time ago, we became interested in trying to understand this problem, and we realized that we could not really understand it unless we knew the forces that cells were exerting on their surrounding tissue and on their neighboring cells in order to close this wound. So we spent a substantial amount of time developing technologies that would allow us to map the forces that cells are exerting. So one of the technologies that we use heavily in the lab is called traction microscopy, that was invented by Yuli Wang and Mika Dembo 20 years ago now. So with Jeff Fredberg and Jim Butler, we generalized it so that we could now apply it to cellular monolayers. And the concept is very simple. So you have a monolayer in this case of cells that is scrolling on top of a soft elastic substrate. We use polar acrylamide, sometimes we use PDMS. So as cells scroll, they generate some forces on the substrate. So if these forces are high enough and the gel is soft enough, then there will be a deformation of the substrate. So we insert these fluorescent nanobits near the surface of this substrate, and then with these beads we can measure displacement field of the substrate, and from this displacement field we can then invert it and compute a traction field, which is ultimately what we're interested in. So I will be presenting a few techniques during my talk, but I will not have time to present all of them in detail. So I refer you to this review that we wrote recently, where we wrote about the techniques that we have developed, but also others have developed in order to measure mechanical stress, in particular in living tissues, rather than at the single cell level. Okay, so with this technology, we were able to turn a movie that originally looked like this into one that now looks like this, where we now have a vector map at every pixel of the movie. So we color-coded these traction vectors according to the sign of the radial component. So those forces that are pointing away from the wound are red. Those forces that are pointing towards the wound are green. And the reason why we decided to use different color codings depending to the sign is because red and green tractions correspond to different mechanisms. So if you look at the movie at the very beginning of closure, most forces at the margin of this wound are red. These are migratory forces, so cells are crawling on the substrate, and for that they generate forces that point away from the wound, whereas at the end forces tend to be greenish, and these are forces that are a consequence of the assembly of a supracellular contractile cable that forms at the periphery of the wound and that contributes to seal the wound through a purse string mechanism. So only by measuring forces, we were able to discern that these two mechanisms were at play and what was the relative contribution of these two mechanisms. And to do so, we resorted to models. So this is a 2D vertex model that we developed, that Vito Conte in my lab developed together with Wayne Broadland in Canada, where they incorporated lamellipodia protrusions for cell migration, as well as a contractile cable at the wound margin. And when they introduced these two components, then we were able to really match not only the dynamics of the system, but also the force fields that we were getting. So I wanted to begin with this because I think it illustrates the way we think in the lab about solving problems. Right? We try to identify an important problem, a fundamental problem in biology. We try to bring as many mechanical variables that we can measure, and then we combine this experimental information with models in order to understand the underlying mechanisms. Okay, so the complementary experiment to the closure of the wound is the expansion of a monolayer, as you see in all these panels. Now, overlaid in pseudocolor on top of each one of these panels is a map of the stress tensor, the trace of the stress tensor in this case. So in the same way that you can measure the forces at the interface between the cells and the substrate, with a few assumptions, you can also infer what is the stress tensor in this monolayer, in this case, assuming that this monolayer behaves as a 2D thin sheet. So each of the panels that you see here corresponds to an experiment in which we knock down one of the proteins of the adosome. The adosome is the complex of proteins that mediate the adhesion between cells. So what we did in this study is to remove them one at a time, and then to observe what was the response in terms of the velocities of the formation of the cells, as well as the forces that they were generating. So with this type of approach, what we were able to do is first to identify which proteins were key in order to transmit forces between cells and to sustain tension in these monolayers. But more interestingly, perhaps, we found some interesting redundancies and feedbacks in the system that one can only address by combining sort of these gene knockdowns with actual measurements of the mechanics of the system. So I'm sure you are all aware that the collective migration of cells, as I've shown in these two cases, depends on the properties of the substrate on which these cells are moving. We showed this, for example, in this case where we had a patch of cells. These are mammary epithelial cells that are migrating on top of a substrate that had a gradient of stiffness. So one of the edges of the monolayer is seeing a soft substrate, whereas the other one is seeing a stiff substrate. What we found is that cells would migrate preferentially towards stiff. This was not surprising. This is called urotaxis. It was also discovered by Yuli Wang about 20 years ago. Now, what was surprising here is that when we prevented cells from forming adhesions with each other, then urotaxis was gone. So in our hands, whenever we try to see urotaxis of single cells, we are unable to do so. When we allow them to join, to hold hands and to form a collective system, then they start being able to urotaxis. So somehow urotaxis in the system is some sort of emergent phenomenon. And through the measurement of traction forces, of tension forces, of substrate displacements, we were able to identify mechanisms at play. And essentially, the crucial ingredient here is that cells need to be able to communicate mechanically from one edge to the other. And this makes them much more sensitive and much more efficient at sensing mechanical gradients. Cell migration is, of course, very important in cancer as well. And for a long time, cancer was thought just as a matter of the tumor itself, which I draw here in green. So it was all about what cancer cells and their mutations do. But there's been this paradigm shift in the field of cancer, in which we now know that the progression of a tumor depends not only on the properties of the tumor, but also on the interaction of tumor cells with cells that are not cancer cells, but that happen to be in the periphery of the tumor or infiltrated inside the tumor. These include leukocytes or dendritic cells or macrophages. These are all immune cells, as well as fibroblasts. And the question that we wanted to address in this study was whether the interaction between cancer cells and the surrounding non-cancer cells was somehow useful to foster the progression of the tumor, or was somehow impairing the progression of the tumor. And what we focused is in the interaction between this fibroblast and the cancer cells. And what Anna Labernadi, who led this study in collaboration with Eric Sahai, she was able to find that indeed these fibroblasts are crucial in this system in order to drive the invasion of cancer cells. So this is one fibroblast that you see here in red, and this is a mass of cancer cells. So I will now play the movie, and you will be able to appreciate that this fibroblast is actually leading the migration of cancer cells. This type of cancer cells, they are epithelial cancer cells. They have very strong adhesions. They have very poor capacity to migrate by themselves. They are surrounded by matrix that they cannot degrade. So it turns out that when they find a non-cancer cell that can migrate, they use this cancer cell in order to escape the tumor. So in this study, we were able to identify a number of mechanisms that were at play. So we found that indeed these fibroblasts are able to degrade the extracellular matrix and to create a tunnel in the tissue. This is something these epithelial cells, so this is squamous cell carcinoma from the skin. These cells are not able to do so, but they are able to hijack a fibroblast that does it for them. And so we were able to show that there is the mechanisms of hijacking by which these cells hijack these fibroblasts are in essence mechanical. And I'll be happy to discuss all these mechanisms later. We'll have plenty of time for it. Okay, so I've told you different aspects of cell migration, which is one of the functions that epithelial cells must be able to perform. But as I said at the beginning, what I wanted to discuss today is integration. Right? So not only how mechanics drives one specific function, in this case migration, but how the multiple functions that epithelial cells must perform, how they are integrated. And Maria Roth, a PhD student in the lab who is now a postdoc in Boston with Christian, she began to address this problem, trying to integrate migration and division. And for that, she used these engineered cells that express this construct called Fucci. So Fucci labels the nucleus of every cell according to the state in the phase that the cell is in. So during the first phase of the cell cycle, these cells are red, then they turn yellow, and during S, G2, and M, they stay yellow. And then after mitosis, they become colorless. So by looking at the color of the nuclei, you can know in what state of the cell cycle one cell is, and you can then also know what is the duration of each one of these phases. So Marina was able to track the movement of each one of these cells for about a couple of days. The cell cycle of these cells is about 24 hours. These are endocyclic cells. So each one of the cells that you see here goes through at least one full cell cycle. So she was able to track the movement of the cells. She was able to track the shape of the cells, the deformations of the cells, deformation rates, and so on and so forth. She was also able to map the traction forces that every cell was generating in the underlying substrate. And she was also able to map tension, which in this case is the trace of the stress tensor in the system. So Marina ended up quantifying for every cell and for every minute of their cycle, she was able to quantify about 20 mechanical variables. And then she was able to ask the question of whether any of these mechanical variables or a combination of them is sufficient to predict the duration of the cell cycle. The answer was yes. And the one variable that appeared to be the key variable was this one at the bottom, the amount of tension that the cell is experiencing from its neighbors. So the higher the tension, the shorter the duration of both G1 and G2. And this was really predictive in the sense that what Marina was looking at was the state of tension during the first phase of G1, so during the first 4 or 5 hours of G1. So if you tell Marina how much stress a cell is subjected to during the first part of G1, then Marina will tell you how long G1 will last and how long SG2 and M will last as well. So this is somehow integrating migration and division, because these stresses that are being generated as the monolayer is expanding, these are stresses that actually regulate the duration of the cell cycle. So there's a feedback loop here that probably cells exploit in ways that we have not discovered yet. All right. So now another function that cells must be able to play in the epithelium is folding, right? So I've been showing you examples of cells that are epithelial that are on flat surfaces, and some of our epithelial are flat, like the skin outside hair follicles is relatively flat, but most epithelial are folded, right? So there is an early Drosophila embryo that is initially has a circular cross-section, and then there's the invagination produced during gastrulation, the highly folded intestinal epithelium that I will be telling you today about. This is a human embryo before implantation, which is spherical, and this is a growing, I think, mammary gland. So most epithelia during development, during homeostasis, are not flat, they are folded. So it's important to incorporate this additional function, which is folding, to this mechanical picture that I'm painting. So the way we did that is from the bottom up, and this was the work of PhD student Ernest Latorre in collaboration with another PhD student, Laura Casares. And on the theory side, we have a fantastic collaboration that's still lasting with Marino Arroyo's group and Sohan Kale, who was a postdoc in Marino's lab at the time. He's now assistant professor at Virginia Tech and a panelist here. So I'm sure that he'll be able to also enter the discussion about what I will be now telling you. So the idea that we had to study curvature was to begin with a soft PDMS gel and micropattern the surface with fibronectin, which is an adhesive protein, but we left some gaps that were not coated. So when we seeded cells on top of the substrate, what we found is, as we would have expected, that cells attach everywhere except for the gaps that we did not coat. If you wait long enough, however, cells manage to cover these gaps. Epithelial cells don't like gaps and open spaces, so they do whatever it takes to cover them. And that's what they did here. If you waited even longer, then they form a hollow dome. so this is actually a very efficient way to generate three-dimensional structure. So this is an array of 20 by 20 of such domes, and if you take a closer look at some of those domes, here's what you find. So these are confocal stacks. This is a top view. And now on the top, you can see a side view along this blue dotted line. So notice that everywhere where we micropattern one of these non-adherent areas, we could find a dome. These domes are variable in their height, and I will later tell you why they are variable in their height. And they are extremely reproducible. Now, what is the mechanism that drives the formation of these domes? So this is driven by ion pumping. So essentially, epithelial, it's always important to think that to keep in mind that epithelial professionals at transporting ions across their surface. And if you control ion transport, you control osmotic pressure. And in the field of soft matter, we all know that controlling osmotic pressure is precious, right? So they control osmotic pressure by pumping ions from the apical surface to the basal surface. This drives a flow of water into the lumen, so they can build up a pressurized lumen. So in our approach, because the substrate is soft, we can measure that pressure by simply looking at the indentation of the substrate. So this is about 3 kilopascal. This is sufficiently soft for this pressure in this lumen to indent it to a measurable extent. So in fact, we did something better than just measure this indentation. We actually used these traction microscopy approaches, but we did that in 3D so that we measured the displacement of every bit in 3 dimensions. And this gave us a 3D traction map at every pixel of the surface of this substrate. So with this, we were able to obtain this type of maps. So the yellow vectors, these are the XY tractions. The dome is here. So of course, under the dome, there are no tractions. There are no shear tractions, as we would have expected. And the Z component of these tractions is color-coded. So blue means indentation, which you observe here inside the dome as a consequence of the pressure inside the dome. And red is a positive force. So these are the forces that are balancing the pressure inside the dome. Perhaps a more intuitive way to look at this problem is from a side view, where you can see how there's a uniform pressure being applied under the dome, and this is balanced by this first ring of cells. So notice that the traction vector here is not parallel to the dome, and this is because there's two components here that cells contribute to. One is, of course, the contribution of the dome. The other one is the contribution of the monolayer, and this gives a resultant that is not always pointing, it is not always parallel to the dome. Sometimes in the domes that I will show that there are gaps. These are not gaps, these are cells that are not labeled. Okay, so these domes are completely sealed. Okay, so now this is really good because you can always fit a spherical cap to this dome. So then you can do very simple mechanics. You can just measure the radius of these domes, and then since you measure pressure and since you measure the radius, then you can compute tension in these monolayers. And not only you can compute tension in these sort of static snapshots, you can actually take advantage of a feature of the system that I did not tell you about yet, which is that these domes are highly dynamic. They inflate, they deflate, sometimes they collapse, then they rebuild again. And this is really good for us because it means that all we have to do is to look at these domes under the microscope. And since they are inflating spontaneously, we can measure pressure, we can measure strain, we can measure the shape of every individual cell, and we can measure tension. So we have all the mechanical information that we want by simply looking at these domes. So sometimes I call this self-rheology because essentially the system is probing itself and all I have to do is watch it. So with this type of approach, we can plot, for example, stress-strain relationships. So on the x-axis here, you see the area strain of these domes. Notice that we're going all the way to 300%. So these are enormous strains. On the y-axis, you have surface tension. So what we found here is that there's an interesting linear increase or sort of linear increase in the first doubling of the size of these domes. When you go beyond that, you have a plateau. And this is a robust result that we have now reproduced many times. These deformations are reversible. So these domes grow and shrink, and if they do it slowly enough, they are completely reversible. And then the other interesting feature of the system is that while these domes always form very nice spherical caps, the shape of the cells is extremely non-homogeneous. So you have cells like the one that I'm pointing at here that is gigantic, and it is surrounded by cells that are very small. This is better appreciated during this deflation event when we took 4 snapshots. So the dome is here inflated 200% in aerial strain. You have 2 cells that are strained 900% and 800%. So I thought that since this is an extreme mechanics letters seminars, there's probably nothing more extreme than stretching 900% if you're a cell, but these cells do it just fine. They don't die. They don't do it. They're completely fine with these enormous strains, which I find it fascinating. And these 2 very cells subject to large strains, they are surrounded by other cells that they barely deform. So one is 200%, the other is 100%. Now, interestingly, this heterogeneity begins about 100% deformation of the dome. So remember that this is the threshold where we start having this plateau in tension, and this is when the variance of cell area starts increasing very, very drastically. All right, so we have a system that has these 2 really interesting features. One is these very large and reversible deformations with a tensional plateau. And the other one is heterogeneous deformation with 2 coexistent phases. So these weakly deformed cells and these super straight cells. And upon seeing these 2 features, Marino Arroyo, our collaborator on this project, realized that this is a feature, that is a characteristic feature of materials that are called super elastic materials. And I'm sure that in the audience, they are experts in this type of materials. The most famous one is this metal alloy nitinol, which I'm sure you're familiar with it, at least you have used it, you have had it in your hands at some point, which you can deform, you can subject it to extreme deformations in a reversible way. And the mechanism is also an heterogeneity in the deformations of, in this case, sort of the atomic domains or the atomic lattice in this system. So in this paper, what we try to do is to establish an analogy between sort of the mechanical instabilities that take place at the atomic lattice level in these type of systems and what we were observing in cells. And so if I wanted to go into the details of this, I would need half more hour. We can go through it in the discussion, but I will prefer to leave it at here for now. Now we're pushing this technology further. So Ariana Marin, student in the lab, is now trying to see how these phenomena scale with the size of the dome. So Ariana found ways to make very large domes, very small domes, and try to see, for example, how the pressure that these cells are able to spontaneously build up varies with curvature. She found that there is a limit of pressure that these cells cannot exceed. They cannot build pressures above this about 100%. She also was able to look at the tension strain relationships depending on the dome side. And what she found essentially is that there are no differences. That what really sets the surface tension in this system takes place at the subcellular scale. In fact, we think that it's just the actin cortex what actually determines this surface tension. Ariana can also make domes that are not spherical. They can be, for example, elliptical. So here you have 2 domes that are ellipses with different eccentricity. We can also inflate them. Now, of course, you cannot apply Laplace's law in these elliptical or ellipsoidal domes, but Sohan Kale, he was able to develop a computational approach which, from the shape of these domes and from the pressure, and without assumption for material properties, he was able to infer the state of stress of every cell in the system. We are also now able to systematically segment each cell in the system. So we are now ready to address questions such as, for example, are cells aligned in the direction of maximum principal stress? This is now much work in progress. It's unsubmitted. It's actually not even written, but this is the directions that we're going in, and I wanted to share it with you and to get your feedback on this. Okay, now to finish my talk, I wanted to focus on a system that I call the ultimate multitasker. and this is intestinal epithelium. The intestinal epithelium is shaped with these finger-like protrusions that are called the villi, and they serve the purpose of packing a very large surface into very small volume. And the key feature of this tissue is its very fast cell renewal capacity. So every 3 to 5 days, all the cells that are lining your gut are gone. They're completely renovated, right? And this happens from the beginning of your lab, of your life, or of your lab as well, and to the day you die, right? And this extremely fast cell renewing process proceeds thanks to the activity of stem cells that are located at the base of crypts. So crypts are invaginations that are located in between the villi, as you can see here. All these are crypts. So crypts contain these stem cells. They also contain Paneth cells. Paneth cells are circulatory cells that I will not be telling you today about. Stem cells in this sort of stem cell compartment, they divide. They move to this transit amplifying zone when they experience more rounds of division. And then they start migrating all the way to the tip of the villus, where they are extruded. And in this journey from the base of the crypt to the tip of the villus, these cells are progressively differentiating from stem cells to differentiated enterocytes and other cell types that are actually enabling the absorption of fluids and nutrients. So during all this process, a cell must be able to perform the following functions: stemness, compartmentalization, folding, proliferation, migration, differentiation, and extrusion. Now, those of you who are sort of in the cell developmental biology field, you will recognize that these are not anecdotal functions in cell biology, right? These are core functions in cell biology. There's conferences dedicated to each one of these functions, right? So so that's why I call them the sort of the ultimate multitaskers, right? Because this cells might be able to perform all these functions. The question that we wanted to ask is, how does each cell know what functions to perform at a given time and location, and to what extent mechanics is playing a role in coordinating this multitasking? And this is the question that was begun to address by 2 PhD students, Carlos Pérez González and Gerardo Ceada, in a collaboration with Daniela Vinovich at Institut Curie. And on the theory side, we still have Marino Arroyo and his group. Okay, so of course, addressing this question in vivo is impossible, right? So you cannot access mechanically the intestine of a mammalian and work on it, right? So it's not accessible. But thankfully, you can use organoids. So I'm sure you are all aware of this revolution in cell biology that tells us that we can now pick just one stem cell in a system like that. And if you provide it with the right physicochemical environment, then you can build an organoid that looks like this, that is somehow shaped like the real tissue. It has crypt-like domains that contain stem cells and Paneth cells, villus-like domains that contain differentiated cells that are extruded into the lumen. So to some extent, they recapitulate, of course, not to the full extent, but to some extent, they recapitulate some of these functions. So we decided to use these organoids in order to address this question about multitasking. And since we like thinking about simple systems, we took these organoids even one step towards simplicity. So we try to open them up and place them on top of a flat surface, and then watch them see if they retain some of the features of this highly folded intestinal epithelium in vivo. So our idea, which was really naive at that time, was to take these three-dimensional organoids, to chop them into pieces, to then drop those pieces on top of our flat polyacrylamide gels coated with collagen 1, laminin 1 with fluorescent beads embedded in, and then hope that these organoids, these pieces of organoids would attach, would adhere, would self-organize, and would form a system that retains some of the features of the real intestinal epithelium. This was a lot of wishful thinking, one of those experiments that you draw on a piece of paper and you think that this will never work. It turns out that Gerardo and Carlos managed to make it work. And this is shown in this movie on the left, where you have two pieces of organoids that we had dumped on these gels a few hours before starting to record. And notice that what happens next is that these cells start to divide, they start to migrate collectively, they start to somehow self-organize, and eventually they form a cohesive monolayer. Let me play this movie again. So you start with these somehow patches of cells, and these cells form this beautiful monolayer where you can distinguish an epithelium clearly, like the ones that I've been showing you before, and some other areas that seem to have a more complicated pattern. So we decided to look at this pattern, and here's what we found. So when you look closer at this area, here's what you find. So this looks like the projection of multiple confocal planes, but it's not. This is just one single confocal plane. And you can appreciate that there are cells with very different shape, which means that there are probably different cell types here. So we use the usual markers to try to see what cells are at play in the system. So we used LGR5 as a marker for intestinal stem cells. We found that these intestinal stem cells are located in this central area here of highly packed cells. All stem cells are located here. Panel cells are also located in these areas, and we find only stem cells and only panel cells. And surrounding this central area with panel cells and stem cells, we found cells that are positive for cytokeratin 20. So these are cells that are differentiated. So these cells are now differentiating into enterocytes and other cell types that now invade the villus domain. So what this told us is that this extremely simplified system retains the main cell types involved in the process of intestinal self-renewing. And not only that, so these cells are compartmentalized in the right way, and they also retain some homeostatic functionalities. This is in fact the first snapshot of a movie in which you can see, if you watch this movie several times, that stem cells at the niche are dividing, they are migrating away from the niche upwards, they spread as they migrate, and eventually when they reach the highest spreading, they are actually extruded into the lumen. This is better appreciated in this other movie where I'm now showing two planes, the plane that I just showed you here on the left, and a more apical plane. So we're imaging higher up now. So notice all these rounding events. So all these are cells that are dividing in the stem cell niche. These are stem cells that are dividing. These cells are drifting radially away. So you can also see all these floating cells. There are cells that have been extruded into the monolayer. If you look at the more basal plane, then you can appreciate all these interesting patterns of cell migration. All right, so this was all very surprising, right? Because if you read your favorite book of tissue engineering or of stem cell biology, you will see that if you want a tissue to behave as it's supposed to behave in vivo, you need to provide it with a three-dimensional shape, with the right ECM, the right in terms of shape, in terms of mechanics, the right ligand density, the right soup. Maybe you will need some cells that are providing the right signals. And here we're doing none of that. So we're doing all the wrong things by the book of tissue engineering, yet cells seem to be doing all the right things, which really calls into question the way we think in the system, in the relationship between what I call these 4 Fs, which is form, function, fate, and force. And this is what we're trying to understand in this system and in others. So in terms of force, we were able to map forces in this system. So this is so the color coding is the same that I showed you for the domes. In fact, the maps are very similar to the ones that I showed you for the domes, even if the system is completely different. So these vectors, these yellow vectors, are the XY forces. They are 0 at the center of the stem cell niche. They increase as you move radially out. They are maximum when you are in this transit amplifying zone where cells divide faster, and then they even invert their sign as they are migrating. And the vertical component is color coded, remember. So the stem cell niche is being pushed towards the substrate, whereas the transit amplifying zone is being pulled away from the substrate. So we took advantage of the fact that this has 360 symmetry. So we average along the circumferential direction. We could average many crypts, and then we could plot the traction force as a function of the distance to the crypt center. We did that for the 2 relevant components in the system. One is the radial component in purple here. The other is the normal component in yellow, in orange here. So notice that the evolution of these two components is complicated. So they change sign. There are spatial derivatives also change sign. But what was really interesting is that these changes in sign, they coincide with changes in function in the system. So if you look, for example, to the normal component, which has its minimum at the center of the stem cell niche and then increases, but when it crosses 0, so when it becomes positive, that's when you have the limit of the stem cell niche, and you enter this transit amplifying zone, and then this peaks and decreases. If you now look at the radial component, it is 0 at the center, as you can see here, and then it drops. It becomes minimum at the transition at the boundary of the stem cell niche, and then it becomes positive. And when it crosses when it becomes positive, then you're at the boundary between the transit amplifying zone and the villus-like domain. So there are very clear mechanical compartments as defined by these force measurements, and these coincide with functional compartments, which suggests that there is a mechanistic link that allows cells to know where they are through mechanics, and this is something that we're actively exploring at present. This is a side view of the same problem. So stem cells are here at the center. So now you can appreciate better the dynamics of the system, how stem cells are continuously pushing towards the substrate, whereas cells at the transit amplifying zone, they are continuously pulling away from the substrate. So this is a complicated mechanical problem, but there is one sort of elephant in the room, if you wish. So there's one very clear feature of what these cells are trying to do. They are continuously indenting the substrate at the stem cell niche, and at the boundary of the stem cell niche, they are pulling upwards. So what this monolayer is trying to do, it is trying to fold. So it's trying to bend, generating a bending moment in order to fold. But it cannot fold. It cannot fold because the substrate is very stiff. These guys are attached to the substrate, and so they cannot fold the substrate. Of course, we reasoned that if we presented softer substrates to the system, then perhaps they would be able to fold. And that's indeed what happened. So we went from 15 kilopascal, which is relatively stiff for these cells. So in this case, the stem cell niche bulges out of this monolayer. But as you go to increasingly soft substrates, then you start having an indentation, and to really, really soft substrates, then you have really a folding that is reminiscent of the folding that we observe in vivo in this system. We can measure tractions in each one of these cases, and we can measure how these tractions depend on the rigidity of the substrate. So rigidity is important in the sense that if the substrate is not soft enough, then the monolayer cannot bend. But beyond that, we wanted to know whether cells are able to sense the rigidity of the substrate and to respond to it. And the answer is yes, but depending on the component that you measure, which is really interesting. The normal traction is independent on the stiffness of the substrate. These cells couldn't care less about the stiffness of the substrate. The radial traction, by contrast, is strongly dependent on the stiffness of the substrate, in the sense that that we think about mechanosensing of, as Mike Sheets probably talked about, which is that the stiffer the substrate, the higher the tension, the higher the traction that cells are generating. So the fact that cells were able to respond to stiffness like this mechanically prompted us to ask whether they are also responding to the stiffness functionally. And the simplest experiment that we did is to see whether the size of the stem cell niche depended on the stiffness of the substrate, and the answer was yes. When we were on very soft substrate, the size of the stem cell niche was large, and it was progressively decreasing as we were going towards stiffer substrates. All right, so mechanics not only drives all these movements and all this generation of all these bending moments, but somehow it also influences functions as important as the size of the stem cell niche in a system like this. Now back to folding. So we became interested in understanding what is the mechanism that drives folding in this system. And of course, developmental biologists have been thinking about folding for decades, if not more than a century. And there's many mechanisms that have been proposed, many mechanisms have been tested and demonstrated, right? So one mechanism that drives folding is bending through myosin differentials, as shown during gastrulation of Drosophila here. So there's an accumulation of myosin on the apical surface of the cells. Contraction of these cells creates this bending moment that that causes the invagination of cells during gastrulation in the system. But you can also fold by buckling, of course, and this was nicely shown by Guillaume Charras in this experiment when they grow a monolayer on top of two cantilevers, and then they bring the two cantilevers together, and when they do that, they apply a compressive stress that then creates this buckling of the monolayer, and then they were able to observe the relaxation of these buckles and so on and so forth. So this is buckling that is driven by an exogenous compressive stress. You can also get buckling in living systems through an endogenous stress, and this is beautifully shown by Aurélien Roux in this paper, where they grow an epithelial monolayer inside an alginate capsule. The capsule is shown in blue here. So these cells, Mdck cells, they divide, they build up a compressive stress, and at some point, this compressive stress relaxes through buckling, which leads to this sort of protrusion here. So you can get buckling through exogenous stresses, through endogenous stresses. You can also fold, of course, through mechanisms associated with ion pumping, as I've been discussing in the first part, in the mid part of my talk. So we piloted many experiments in trying to understand which of these mechanisms was at play, and all of them had been alluded to, discussed in the literature, or attributed in the literature as mechanisms that could explain bending in the intestinal epithelium. So I will just give you a few of those experiments. So one that is what was a really obvious experiment is to inhibit contractility in the system, and this is very easy to do. You add blebbistatin to your cells, and then what we found is that all forces are gone. That is expectable, but folding is also gone. So this was telling us that whatever is driving folding in the system, this was driven by myosin. So if this was the case, we better look at actin and myosin in the system. I'm showing you actin, and myosin shows similar patterns. So we looked at the localization of actin in the system. So we found that actin is located at the apical surface of stem cells. This is something that was already known before us. But we also found that actin was forming a ring surrounding the stem cell niche in the basal, sort of the bottom surface of these cells. And so this again raised the possibility of two mechanisms. One was this idea of apical constriction, right? Cells at the top contract, and this creates this bending moment. But the other possibility was that these cells would form a contractile ring that would compress the stem cell niche, and then the stem cell niche would then buckle as a consequence of that compression. So we would have basal compression of transit amplifying cells. Now, to differentiate between these two processes, all we needed to know is to access the sign of the stress tensor in the system, right? If the system was buckling, then we should observe compressive stresses. If it was bending, we should sort of see tensile stresses. So for that, the best technology that you can use in mechanobiology is laser ablation. And that's what Carlos did. He used a laser to cut the monolayer in different regions. So when he cut in between the transit amplifying zone and the villus-like domain, here's what he found. So let me play the movie. So the cut is this red line. So I will play the movie a couple of times. So you can appreciate that when he cuts the monolayer retracts in both directions away from the cut. So this means that the system is under tension. When you cut at the interface between the stem cell compartment and the transit amplifying zone, you observe the exact same thing. So you cut here, and then again, cells recoil away from the cut. Let me play this again. Okay, so we did these experiments in many crypts. We quantified that very carefully, and we found that all the time the system is under tension, which rules out this idea there is a compressive stress that buckles the system. We don't think that is the case. We think instead that this apical constriction is really what drives bending and folding in the system. There's a parallel paper. This was published recently in Nature Cell Biology. There's a accompanying paper by Prisca Liberali and Eduard Janezo, where they reached the exact same conclusion with three-dimensional organoids. So we wanted to push this study a bit further, because besides the mechanisms that drive folding in the system, there's another elephant in the room, if you wish, which is the extreme heterogeneity in cell shapes that you can see in the system, right? So there are some cells that are extremely elongated, some other cells that are cuboidal, tall, thin. So we thought with Marino, well, can we explain in simple mechanical terms how all these cells with such different shapes coexist? And for that, Marino and Francesco, a postdoc in Marino's lab, develop a 3-vertex model, and many of you in the audience in the panel are experts in this type of models, of the monolayer, in which they start with a sort of a naive epithelium where cells are identical. And the one thing that they did is to prescribe surface tensions in the system that would follow the profile of actin, of cortical actin in the system. So in other words, we measure cortical actin in the system and we prescribe tensions that are following that profile. And by just doing that, here's what they found. So they found that there is apical constriction in the system, and they found that there is some very pronounced changes of shape as you move away from the center of the stem cell niche. So Maria Matejcik, a postdoc in my lab, measured these experimental crypts and segmented them very carefully. And then she was able to compare from a morphometric point of view the predictions of the model and the actual data. And she found that the match, with very little assumptions on the model side, really, this was really straightforward. The model didn't really need much tweaking. In fact, the more we tweaked, the worse it was getting. So cells are taller at the niche. As you move away from the niche, you see this tilting. Then there is a region where we have these banana-shaped cells, much as the ones that are predicted here by the model. We see some basal constriction, and then cells become cuboidal as we move towards the villus-like domain. So what these experiments were telling us is that by simply knowing, so that all cells need to do essentially to explain this extreme heterogeneity of shapes is to tune the amount of actomyosin that they have in their cortex, which I think is an extremely powerful result and a very simple resource that cells have in hand. All right, so with that, I finish here. I want to acknowledge everyone who did the work. I mentioned their names as I was going through my presentation. This is my current lab. Collaborators, Marino Roy and his team at UPC in Barcelona. Sohan Ker was at the UPC when we did a lot of these studies, now at Virginia Tech. Daniela Ensa-Ducurie, Daniela Vinivich, and her team were super helpful in all this intestinal epithelium stuff. They taught us everything we know about the intestinal epithelium. Collaborators at Mdck, Pera Roca, our technicians at IRB, Edu Valle. I thank you for your attention. These are the funding agencies, and I'll be happy to discuss about anything that I showed you or anything related to sort of, I guess, sort of more general epithelial mechanics. Thank you.
Thank you very much, Xavi. I'm gonna clap on behalf of everybody here. I think Jimmy usually asks the presenter to stop sharing the screen so that we can see all the panelists. So, like this also, please all the panelists turn on your video so that we'll see all of you. And then in order to ask questions, both the panelists and attendees, please use the raise hand feature on Zoom. And then, I'm gonna call you. And then when we ask questions, I'll also ask you to just please briefly introduce yourself, say your name and your affiliation, and then we're gonna proceed. So I received the first raised hand by Daniel Cohen. Please go ahead, Daniel.
Hey, Xavi, that was great. Gorgeous, as always. Good to see you again. I have a sort of goofy question to kick off, but you show these 900% strains and these cells are clearly extreme. What is the limit of extensional strain for an epithelial cell? And in terms of in vitro experiments, and then what is the actual maximum that you think happens in vivo?
Well, so they have a nucleus that they cannot compress. It's too stiff for them. So this is, I guess, setting one of the limits. We're now trying to, instead of allowing the cells to stretch by themselves, we're trying to inflate them, so that sort of to pump water into them and to really blow this, this dome and see what is the limit. Because what we're seeing is that these cells are able to soften their cortex substantially, so they can really dilute their actin cortex. And then what really governs their mechanics is the intermediate filaments. So, but intermediate filaments are also to some extent stretchable, right? So 900% is what we are where we are right now. How far can we go? I don't know. And in vivo, every time when I used to give talks in person, at the end of my talks, there was always someone who would come and say, I know this developmental system in which cells become huge because they deform, and you should look at this organoid, these other organoid, like bladder, for example, is one of these systems in which you would expect that cells are able to undergo massive deformation. There's a lot of folding as well, no? But at some point, so I'm looking forward to hear from people from sort of more dev bio or physiology to know what are the limits of strain. So since they can dilute their cortex, and because they reach this plateau of tension, then stresses don't increase so much, so they are able to deform substantially.
Nifty.
Without breaking junctions.
Yeah. Thank you, Xavier. And maybe just a comment, like for the panelists, if you have a follow-up comment on the question that, or like the comment that Xavier just raised, please, just unmute yourself and, add additional perspective from your side. So next we have Li Qing.
Could I?
Okay,
sorry,
go ahead, Jeremy. Yeah.
Perhaps I can just sort of jump in and ask a sort of very closely related question. And sorry to interrupt, but Xavier, again, lovely to hear this stuff. So I was just reflecting on, one of the reasons why they can extend their area so much, of course, is they're getting very thin that way. And as you mentioned, the nucleus is a bit incompressible. Do you have any insight into whether cells, other than with the nucleus, do anything where they have stiffening that tries to maintain height? Because, in real tissues, in all of these epithelia, there's always a characteristic aspect ratio, right? Mm-hmm. And, there are two sort of schools of thought, if you like. There's one which is that it's a balance of tensions at the cortex, and that's the sort of vertex point of view. With obviously some turgor pressure to maintain that. But then there's also kind of hints of things like microtubules and other stuff that might also be helping to push the height. So I'm going to ask this question in a sort of very vague way, but have you sort of thought about that or looked at that or get any hints that there's something other than the nucleus that's keeping these cells not four lipid monolayers thick?
Right. Exactly. Yeah, I guess there would be other organelles, right? But so I mean, these are MDCK cells. They are not very polarized in the system. They have some polarity, but I don't think these cells have lots of microtubules there that would be sort of generating some sort of trying to keep up this height, right? Which I'm sure in other systems happens, right? So one thing we would love to do, so when we look at these cells, they are super thin. They are really thin. I don't know if it's like 4 bilayers of membrane, but they're really, really, really thin. And they can extend these very long protrusions that are very, very thin. But we want to put in there some cells that are more realistic. Maybe these organoid cells that I was discussing, some cells that really, for which maintaining polarity is really important and maintaining height is really important. And this we would love to stretch. And as I said, then you need a system in which you can indeed control the stretch on your own, which is something that we're working on, but we don't have ready. But this is a very good point that I'll get back to you.
Okay, thank you.
All right, now let's go back to Li Qing.
Thank you, Professor. Yeah, actually, at the beginning, I introduced myself. I'm working in Paris, but now I'm traveling in Singapore. So, this topic is very interesting. I'm very, interested and learn a lot from it. So here from the talk, I just have 2 simple questions. So when we deform the cell structure or stretch this kind of structure, the deformation, all the deformation is only observed by the cell themselves, or could be also part of the deformation could be observed by the gels or the material between cells. Another question is about, could we, if it is possible for us to observe the cracks or deconformal between the cells when we stress them? Thank you.
Right. Yeah, two good questions. So, I mean, here essentially the system in which you don't have ECM in these domes. So all you have is cells. There's no material in between them. So it's just a tight pack of cells that are stretching and deforming. So all the deformation is taken up by the cells. And with regard to the point about leaks, this is a very interesting question that we wanted to address, but as everybody, I mean, there's people here who have been working on fracture for a long time, right? And to know where fracture will happen and when fracture will happen is very hard. And here you cannot image the cells at a very fast frequency for a very long time and just wait for a fracture event to happen and then observe some sort of a jet of fluid that leaves. Or this would be, so of course you can provoke leaks, and we've done that. So you blow a small hole with a laser, and then you see that boom, the dome collapses. But to catch it is very hard. And even if we were able to catch it, the size of these leaks must be quite small. So I don't think we can resolve them with optical microscopy, at least with the optical microscopy that we use. So I think it will be very interesting to understand the fracture mechanics of this system, because of course, fracture mechanics of epithelia is a super important problem in physiology, right? If you get leaks in a tissue, then it's a problem, or maybe it's not a problem if you're in the kidney, no? If you're in the kidney, that's what you want. If you are in other tissues, you don't want leaks, no? So understanding the fracture mechanics of epithelia is super interesting. But I don't know how to do that. It's just too rare events for us to sort of be sitting there and observe. It's just too rare, too small.
Yeah, thank you. Thank you. Very good talk. Thank you, Mr. Hu.
All right, let's go to Daniel Matos.
Hi, Daniel Matos. I am assistant professor at the University of Warsaw. Just introduce myself again. Yes, I have a question about your last experiments about the organoids. I'm a little bit puzzled about that. You said that you observed the same function somehow when you flatten them down, right? So I'm a little bit puzzled about that, because I would think that the curvature in some sense is important for this type of organoids, right? And what you are saying is counterintuitive in some sense, because you are saying, well, if I just flatten down this, is actually the form and the function are separated, right? So do you have any insight about that? Because I would have thought that actually the curvature is very important for the intestinal organoids, right? So I'm puzzled about that. So I don't know.
I'm still puzzled. I'm still puzzled to accept that sort of evidence and puzzles you. But you're completely right? Because it's a bit what I was saying, that this is really against the way I think about mechanobiology, because I know that curvature should be important, right? But it's not in this case, right? So there's another lab, the lab of Matthias Luthoff in Lausanne, who's doing some experiments where he finds that stem cells accumulate in the regions that have higher curvature. So he imposes curvature on the substrate. And then he finds that indeed these intestinal stem cells, they localize in the regions of higher curvature. What we show is that you don't need curvature at all for these stem cell niches to form. So I think that what happens is that spontaneously there's some sort of signaling in the system, or there are some sort of rules that spontaneously there are some fluctuations that become amplified, and the system patterns. So there's patterning, and there's a patterning of these scripts. If you impose curvature, this tilts the balance towards having the crypts located in the script. So it's not that curvature doesn't matter, at least according to the talks that I heard recently from Matthias, and I think he has a paper to appear on Science about this, as he mentioned. So probably curvature is a cue that favors patterning in the system, but you do not need it. It is not a requirement.
I guess my second question about that is, what are the physiological cues are you base it to say that are performing exactly the same function? Because there's
a number of chemical signals, wind, BMP, So there's gradients of efferents. So there's a number of ligands that are very well studied in the system. And if you pattern surfaces with these gradients, you can actually also control the formation of crypts. So there's sources of these signals in vivo. So not in the epithelium, but in cells that are surrounding the epithelium, that are sources of these ligands that sort of help patterning as well. So there's so in other words, it's a very redundant system in which chemical cues can do it, and physical cues can do it too.
And what about if these time scales are really separated in between both of them, and actually you are not accessing them? So in the sense that essentially what you are pruning in your experiment are just the biochemical cues rather than the mechanical cues, the ones that are responding in the span of your experiment. It can be
so what are the time scales? I don't know. I think no one knows. It's doable though. I mean, one can try to micro-pattern these ligands, or to add these ligands and see how long it takes for these systems to form and to sustain. To differentiate into intestinal stem cells. So I think these are very pertinent questions. I don't have an answer.
Thank you.
I have a follow-up question.
Go ahead, Noah.
So Matej from Microscopy Institute. Okay. So about curvature, right? So I always thought that at the tip of the villus where cells are extruding, that they are extruding easier because they're already a bit basally constricted, right? So now when you extend it to the flat surface, these cells are now completely flat, right? So they're no longer neither apically nor basally constricted. So, and in contrary, there are cells where the villus and the crypt region comes together at the rim of the crypt, like you showed, right? Where these cells in the real structure, they're flat, but in your structure, in extended structure, they're basally constricted. So do you see any differences in extrusion, amount of extrusion in these two regions?
Yeah.
Comparing your system and the real system.
Yeah, you're completely right. So when you look at the real gut, and there's beautiful papers by, for example, Daniela Minevich at Curie on this, cells at the tip of the villus, they are more packed, so density is higher, and there is some basal constriction, which I also thought, well, as a physicist, we like physical variables controlling everything, right? So I said, well, that's cool because there's higher density here, and this will be the signal that will trigger extrusion, right? Too much density, maybe that's compression of some sort, and boom. And now we find in our system, the cells that are extruded are the ones that have the highest spreading area. Yet they are also being extruded just fine, right? And so this is also very puzzling, and it tells you again a bit the same thing that I was telling you. I'm sure that the fact that they are more compressed and maybe some sort of basally constricted helps extrusion, but you don't need it. You don't need it at all for these cells to be extruded. You can have them extruded under conditions of extreme spreading. Yeah.
Okay, thanks.
Yeah. Thanks.
Go to Noah Mitchell now.
So I had a quick follow-up question to Daniel, and then that's maybe more specific, and then a more general question. So the follow-up would be, so in the experiments where you varied the stiffness of the substrate and you saw more curvature in the crypt for low stiffness of the PDMS. So there you showed that the radial force was variable with the stiffness, whereas the tangential, Z force, the traction in the Z direction was not, right? It was invariant. And so in this radial case, are you using the the reference state of a planar configuration to define that radial direction, or is it the local tangential frame of the curved final state?
Yeah, we checked that carefully, and And we found that essentially this mechanosensing was present, regardless of which, of course, there is a variation, right? If you project on the curved surface, or we take the frame of reference of X, Y, Z, right? But the changes were not significant in terms of this mechanosensing. So it doesn't really matter. So at the end, I think we reported everything correctly, taking into account the substrate rather than the XYZ normal axis.
Yeah. So I guess I mean the question there is kind of, what do you make of that sensitivity to the curvature? I mean, is it a sensitivity to curvature, or is it a sensitivity to like sort of extracellular matrix stiffness, right? Like, do you feel that this is the material responding to its ability to change shape, or do you feel that this is the tissue to change shape, or is it the tissue responding to its direct interaction with the substrate? Do you understand what I'm asking?
Yeah, I think we see so for example, if we look at mechanosensing, so the changes are very big. The changes in curvature are relatively small. So if we are in the regime, so if we focus on a regime that is unphysiological, but so you go from 15 kilopascal to maybe 1 kilopascal, the folding is not that pronounced. So you're still pretty much unfolded, and you see these drastic effects already. So I think it's not the curvature. I think it's the stiffness in this case.
It's the stiffness. So then, so then the more open-ended question is, I think it would be really, really cool to see, I mean, if you were able to have a bilayer physiologically, like you had some mesodermal layer that is like what's present in the intestines and in vivo, we have both muscle layer and endoderm, and there's this interaction which has been widely, I mean, also probed in different studies as well. And so, yeah, I guess is that a direction that you see as feasible in vitro, and how do you sort of configure your results in the context of the mechanics that could be imbued upon the epithelium from the mesodermal layer?
So you mean to start with not already chunks of organoids, but start some sort of a more fetal-like state where you can observe the more of developmental evolution rather than homeostatic. Is that what you?
I suppose so. I actually don't know. Is it possible to have a bilayer tissue with both differentiation of mesoderm and endoderm in an organoid? I'm not sure. I mean, that's an open question in my book.
I don't have an answer to that. I'm sorry.
No, it's okay.
I mean, maybe there's someone around who does.
But I guess either way,
I don't know.
Yeah, I mean, I guess either way, like, would it be possible to sort of engineer some sort of differentiated musculature or myocyte-like cells and add your crits on top of them? Would they, adhere to that extracellular matrix secreted by myocytes?
I think we can build up on that. Because I think the hardest part is already done. The fact that we can unfold one that must be the most folded tissue in our body. We can unfold it and it is still functional. And then if you want to do that on top of a system that on a layer of muscle or of some sort of other type of tissues, I think that would be perfectly doable. And maybe we should do it.
I'm sorry, just a point. And didn't Daniela do some of that? With some, I forgot what. My
she might be growing.
Taking the gel.
Right. I think she. I don't know if she's published that. I think I've seen data. I don't know if probably from Daniela or from other people.
I think she did.
They grow monolayers of fibroblasts. or mesenchyme. And then on top of that, they add the epithelium in order to understand the crosstalk, because so I told you something that is very epithelial centered, right? But when I show you these beautiful images of this highly folded system, there's many other cell types in there, right? So there's at the base of the crypt, it is wrapped around by I mean, there's fibroblasts, there's muscle. So I mean, the cool thing is that if you don't have any of these other cell types, so for example, I thought that that to sustain this curvature, you would need muscle. No? So I thought that, well, maybe epithelium contributes to bending, but what really drives and sustains the tissue, this is actually the muscle that wraps around these crypts, right? And that's probably happening. I mean, it is happening because we have the images of that in vivo. But when we look at Daniela's data, because we shared lots of data, we found that the contribution of the epithelium in sustaining the shape is very important. In fact, for example, when Daniela, when she knocks out myosin in the epithelium only, the shape of the villus of the crypt is lost, which is very surprising, meaning that even during homeostasis, there's still a mechanical contribution of the epithelium. So we tend to think often about the epithelium as playing very important role in development, in driving morphogenetic movements in development, but then to be sort of passively sitting on top of the ECM, where tougher cells like smooth muscle are the ones that sculpt the tissue and sustain. And maybe that's not so clear. I mean, part of it is for sure is that, right? But I think the epithelium is playing an actively mechanical role in sustaining the shape of these tissues.
Thank you.
Thanks.
Let's move on to Hai Chao.
Hi, Professor Trepat. This is a very intriguing talk. I'm Hai Chao Han from UT San Antonio. So I have two questions. One is more about the dome, the 3D dome experiment. So I may miss the detail. Is that you have a very locally a circular base, a membrane, right? You culture, it's very soft, so the cell will grow and then they will buckle or somehow deform into the dome shape?
Right. No. So there's no buckling in the system, and which is a pity because it would look really nice. But so far there is no buckle. We're trying to engineering buckling in the system, but basically what you have is a layer that has differential adhesion. So there are some areas that are strongly adhered to the substrate, and some areas that are sort of floppy, not really adhered to the substrate. So then these epithelial cells start pumping ions across their apical surface. In the regions where cells are strongly attached to the substrate, the osmotic force that you build up is not sufficient to detach, to delaminate the monolayer. But in those regions where the adhesion between the cell and the substrate is weak, then this osmotic pressure is sufficient to delaminate the monolayer and start building the dome. And then you keep pumping ions, and you start building up higher and higher pressures. And what was really cool and hard in these experiments was to find the balance between adhesion with the substrate so that we could delaminate one part of the monolayer and not the other part of the monolayer. And the fact that we can control this so precisely is really cool and to some extent also unexpected.
Okay. So the pumping is done by the cells, or is you are pumping?
No, it's done by the cells.
Okay.
So these are kidney cells, and that's what they do.
So the cell will pumping liquid inside, and then they grow kind of more bigger, or they can change, right?
Well, the cells cannot pump liquid; they can pump ions, and that drives liquid through cell-cell junctions and through the cell membranes.
So it's because the fluid get in, and so that deform into the dome.
Yeah,
thank you.
It's water coming from outside.
Thank you. My next question is, so for the epithelial cell, it's about your first experiment, the wound healing experiment. So for the epithelial cell, are they sensitive to flow? So what if you have a flow on the surface? Will that affect the healing pattern, where it move, or the fluid doesn't matter?
The flow of the liquid that's on top of them?
Yeah, on the top, on the surface.
Yeah.
If you add the flow, would that affect it or no?
I'm sure it would. The effect would be bigger on endothelial cells, but probably also in Mdck cells. I mean, there's papers out there. There's papers about it probably also in organoids. So we have ignored that. So not that we have ignored it. We don't add that component. And we have a paper. So we have a paper on endothelial cells where we add flow, and when we add flow, forces increase quite dramatically. And this is something that has been reproduced in several labs, I think. In these systems, the systems I showed today, I don't know, but they are sensitive to flow. And for sure.
So
again, there are experiments in the literature. They are very old experiments to apply flow, and you deflate the blisters with the flow.
Okay. Yeah,
they are saying it's because of the celiac.
Oh.
I don't know if you know. I just came across this paper.
Yeah.
No,
so what Jack is pointing at, and it's worth mentioning, is that these domes that I presented, they were observed for the first time in the 50s. So there's a science paper in Mdck where they find this, and they relate it to cancer and stuff. But there was a series of very clever papers in the 20 years or 30 years after that they really use these domes for a lot of experiments about transport of ions. And there's really clever papers about transport and about mechanics. And then people started doing more complicated stuff like cysts, full cysts, organoids, and then all these domes were abandoned. I mean, the domes that they were planned were spontaneous, that they were in these papers were spontaneous domes. Sometimes these domes were spontaneously. So we took these very old papers and then applied the micropatterning technology in order to control their size and their shape, which now allows us to, but as Jack was pointing out, there's all these super clever papers, 2 figures, and that you read and it's like, wow, holy shit. So it's a very nice literature.
Can I ask some more questions about the domes? Is there a coupling between the pumping and the mechanics, or are the cells pumping anyway?
We don't know, because so we attribute so the domes are very dynamic, which we attribute to leaks. But it could be as well that there is some control, right? So that as the domes inflate, and then they stop pumping, and then that's why they deflate. So there probably is some coupling between pumping and stretch, right? It would be hard to imagine that these pumps that are sitting on the membrane, you stretch the membrane by this enormous amount, and they can continue to work without some sort of feedback, right? So that's something that would be interesting to study. Maybe we would have to find ways to measure that. Maybe try to build up domes and come up with ways to keep their volume constant, and then sort of, or I don't know. It's not easy experimentally to address that question. I don't know if you have any idea how to do that.
Another question I had is, you talked about super elasticity. Could you say more about that? For instance, are these weakly deformed cells and the super stretched cells completely mixed, or do they organize into whatever? Do you have a qualitative explanation of why this is super elastic?
No. Well, yes, we do have we do have an entire theory on it, but that I didn't get into it because it was just too much. But so our idea is that so as these domes inflate, as you know, they have a surface tension. Every cell has a surface tension that arises from the cortex, and we know that from your work mostly. But this, as you stretch the cells, they manage to have a cortex that continues to have the same density of cortex, and this is because you have a ton of actin monomers that are ready to be polymerized and to be sent to the cortex, right? So our measurements are consistent with the fact that as the dome begins to stretch, the thickness of the cortex is relatively constant, right? But of course, at some point, you will run out of actin monomers, and you will no longer be able to have a cortex. that has the same thickness, and therefore you cannot no longer have a constant surface tension in the system. So then the first cell that reaches that limit, that cell will begin to soften, right? Because the system continues to stretch. You're being pulled by your neighbors, but you're out of monomers of actin, so you cannot build up a cortex anymore. So cells start pulling on you, and this is an instability, right? So because you're softer and softer, you're more stretched because you're softer, and then your neighbors start pulling on you. So eventually, there is one cell, the first cell that runs out of these monomers in the cytoplasm, that starts to become stretched. So that's why we think that the system, that the behavior that we observe is biphasic, right? That you stretch, and then one cell, boom, pops up because of this instability, right? And then once, but then that one cell that becomes unstable, that cell should take up all the strain, right? And that doesn't happen. It doesn't happen because the intermediate filaments in the system, they then take over the actin cortex, and they are the ones that prevent that cell from stretching further. So that cell reaches a limit of stretching, and then there's another cell that gets into this instability and then pops up. So at the end, you have all sorts of cells that start popping up, and that's why you have the coexistence of cells that are super stretched and cells that are not stretched. So I don't know if that was clear enough.
So are there arches of superstretched cells or chains of superstretched cells or not?
So that we want to look at. So, but again, the experiment that we need to do for that is to really inflate in a controlled way. So what happens in this problem is that since we have all these fluctuations superimposed to the system, we don't know well the history of every cell in the dome. So we don't know if we repeat this experiment twice, is the same cell, the one that becomes super stretched or not. So what we really want to do is to be able to inflate these domes in a controlled way. So we know the history of every cell in the system. And then we can monitor who gets super stretched and whether there are chains that form in there, whether every cell at the end becomes super stretched. So what are the limits, right? And this is very hard to do in our system as it is right now. But hopefully we'll be able to look at that in the future.
Sorry, can I, Xavier, can I just very quickly, sorry to jump in, Jeremy Green, King's College London, sort of a question and a suggestion, which is, have you used any sort of channel blocking inhibitors to maybe turn the tap on some of these transporters?
Right. No, we have not. There are, so you can foster.
Might be able to, you might be able to modulate the rate of inflation, and maybe.
Yeah, we can do that. We could do that.
Just a thought.
Yeah. No, that's. I mean, they are well characterized. They have been used in these systems, so that's something that we could do. So we can accelerate the rate at which inflation happens, or slow it down. So these are experiments that can be done and that we have not done.
I had some related questions to the dome, and I was wondering if I can go next. So this is Momita Das from Rochester. So my questions were, one is that, so your cells are Mdck cells you mentioned. And I also understood that no matter what is the length scale associated with the gap, you always get this dome, right? It's never like undulations or something else. So one thing I was wondering is, instead of Mdck cells, if you had something like fibroblasts, and if they secrete fibronectin and you have a much stronger sort of elastic, might you see a different kind of structure? That's one question. And the other question is, if you sort of could introduce artificial localized concentration gradients in your, in your ions, right, and the salt concentration, can that again lead to some kind of different kind of structure instead of, just a dome as you go to different length scales for your this gap?
Well, the second is sort of one of the dream experiments in the system is to find optogenetic control of pumping. And to my knowledge, this doesn't exist. But if, I mean, we've developed optogenetic tools for contractility, but I really don't want to start developing optogenetic tools for pumping. But if someone does it, that would make me really, really happy, because then you can have one cell pumping like crazy and others not. Gradients, right? I mean, wouldn't that be fantastic? We could sculpt shapes as we would like. So it would be very, very nice to have this tool. I don't know if anyone knows who can do that.
Well, I asked Virgil, I asked Virgil Vyasnov to do some of that.
Yeah.
And what he tells me is that you can find channels, but not pumps. So you can, yeah.
Okay. Right. I mean, these pumps are complicated, right? So they are complicated machines. Yeah. But I think the transport people should do that because there's no methods. And I don't know, maybe we will end up having to do it. But so far we're trying to escape. And then from the other question, so I mean, fibroblasts, so they don't pump. They are not so cohesive, so we cannot use them in the system. If we had a sort of universal inflator where we could control the differences in pressure, then that experiment would make a lot of sense, right? And but trying to turn this into system that can actually buckle, that's something that would be lovely, right? That would be very nice to do, and it's on the list.
Thank you.
Sorry to jump in, but Xavi, we have weird data with MDCK cysts and domes that we can cause them to inflate on command with electrical stimulation, which is thought to control the ion channels. I don't know if that's useful. We can talk offline. We're still trying to find the mechanism, but we might be able to actually turn inflation on and off. And we're trying to think more about the mechanics there. So maybe I'll bug you later about that. But
okay,
it's a weird system, but it does control swelling.
Okay, yeah, that would be great. I mean, that would be great. A switch to inflate, to turn on inflation. Okay, so we should talk about this. Absolutely.
Right.
Yeah.
Thanks.
So in the chat, I saw a question from Kaoru, which was like a follow-up question to your remark, Xavi, about Jean-Francois' question on superelasticity. Kaoru, do you want to go ahead and answer?
Okay, so I'm Kaoru Sugimura from University of Tokyo. So to address the monomer depletion hypothesis, have you looked at how changing actin turnover and actin assembly affect the cell area heterogeneity?
Not actin turnover. Well, we used an inhibitor for ARP2/3,
okay,
and it didn't do anything remarkable in the system. We did not measure dynamics of the cortex. We did measure the density of the cortex, I mean the thickness of the cortex and how that varies as we are inflating. But not the dynamics, like the turnover rates. We just assume them to be the ones that govern actin polymerization that have been developed by found by others. Yeah.
Okay, thank you.
Thanks.
Right, let's go to Kinjal.
Yeah, hello. Thanks for the great talk. This is Kinjal Das Biswas from University of California, Merced. So my question is about the different strategies to study folding and possibly buckling, and also the different strategies that cells might employ to do that. So one specific question I had is, I presume that you are doing these experiments on thick gel substrates because you want to measure the traction forces. But is it possible to also do it on thin substrates so that the substrate might also buckle with the cell, so that the cells do not have to de-adhere from the substrate in order to go out of plane? And we know that thinner sheets are more easier to bend and buckle. So
right,
would that be a strategy to vary the thickness of that?
Right. So these substrates are 50 to 100 microns in thickness, but they are attached to a glass at the bottom. So that's why they cannot buckle. So I guess what you're saying is to do this experiment, but without having the soft gel attached at the bottom to a glass, so that then you could have a fight between the pressures and the tensions in the dome and the substrate, and therefore the substrate could buckle.
Right, and I'm not an expert, but wouldn't that also be closer to the in vivo situation where there's a thin basement membrane and not a thick substrate?
Well, yes, there is a, well, depending on what condition you're trying to mimic, no? But indeed, the basement membrane is a very thin and very stiff material, whereas what we do is something that is thick and soft. So But there's a lot of mesenchyme in ECM that is sort of more diluted and thicker. No? But you're right. So if what you're trying to understand is these dynamics of the relationship between the epithelium and the extracellular and the basement membrane, but in that case, you would hardly detach the basement membrane from the epithelium, right? So they are usually very closely attached. But indeed, if there is some detachment because there's some blistering of some sort, then that would I agree with what you're saying is realistically, and it would be really nice to see what the substrate would do.
Okay, and can I ask a more general question related to that? And is that like most of the mechanisms I have seen, and that you also verified with your laser ablation, is that the out of plane or the bending is indeed driven by the like the actomyosin contractility on the apical surface. But are there other possible ways of buckling that people have, or of folding that people have seen in these epithelia? Like, for example, one simple way for thin sheets to buckle is to have differential stresses. So if cells in one region, let's say, were expanding more in plane than in the other, that could also cause buckling. But I don't know if that happens in biology. So
it does happen in biology. Buckling happens in biology. And it's been engineered by Guillaume Charas, by Aurélien Roux. So you can engineer buckling exactly how you said, right? So by cells having like differential growth, this has been engineered. And there are developmental processes that have been attributed to this, to buckling. So I think that that's actually a mechanism that is used in certainly used. I don't think it's used in I mean, it's only used in physiology. I don't think it is in, or evidence that argues against it in the gut, in the folding of the epithelium of the gut, not in the morphogenesis of the villus, for example. In fact, the formation of the villus, not of the crypts, but of the villus, is one of the processes that has been, I think, quite convincingly shown to be caused by buckling.
No, okay, thank you.
Mm-hmm. All right, let's go to Z-Chat.
Thank you. Thank you, Jack, for the wonderful talk. I have a couple of questions. First is, to what extent can the cell volume change during collective cell migration?
To what extent? Sorry, I missed the first part.
Oh, I was wondering to what extent the cell volume could change during collective cell migration.
It's yeah, I mean, you bring up a very interesting question, the question of, and I think it's a controversial one as well, no, of volume conservation or not. I mean, and so the person who knows more about this is Mathieu Piel at Curie, who has a wonderful preprint on volume regulation. And Ming Wu also at MIT has been looking at that, and other people in the past, of course. So what measuring volume well is not so easy, but when cells spread, and so basically when you deform, you change volume, depending on the rate at which you deform. And this is what Mathieu Piel convinced me in his most recent preprint, and I think this is a super interesting phenomenon. We do see that at the leading edge, cells are much more spread and they seem to have less volume. In the domes, when the cells deform so much, we think that there might also be volume changes, but we have not been able to quantify that carefully enough. So I don't know.
Yeah. All right, thanks. Sorry, I forgot to introduce myself. This is Zi Chen from Brigham and Women's Hospital and Harvard Medical School. So my second question is, so in terms of the mechanisms of force generation, are there any differences between, collective cell migration during wound healing versus morphogenesis or cancer cell migration?
Well, there are similarities and differences. I mean, one idea is that cancer hijacks the mechanisms of development, right? So that's one. I think that is one beautiful idea, right? That cancer cells continuously find ways to hijack what cells do during development, to use it for invasion, for metastasis, for growth, and so on and so forth. So at the end, many of these mechanisms are recapitulated in terms of both amoeboid migration or protrusive migration, in terms of degradation of the ECM, in terms of adhesion between cells. So I think there's lots of similarities. So I would say that these mechanisms are very, very analogous.
Thank you. Let's go to Jose Munoz.
Hello, hello, Xavi. I'm Jose Munoz from Barcelona, also at UPC in Spain. And I'm curious, a little bit of a change, because it's about some years ago, there was the say that when you have drastic morphogenetic changes, like lots of remodeling, you don't proliferate, or you proliferate when you are more in a stable position. I think that what you showed of Marino Rose, showing which is your cell cycle related with tension, maybe answering this a little bit, saying maybe it's not either you proliferate or not when you have drastic changes, but when you have tension, okay? I don't know if you agree on that, whether you have lots of, you see it correlates really morphogenetic movements with proliferation, or it's not necessary so, or would you
right.
still?
Yeah, I think there's this is a comment that I've heard several times, that somehow when you move, you don't divide, and when you divide, you don't move. And in my experience, that is not true. And it's very clear in this case of the intestinal epithelium undergoing homeostasis, that you see these cells migrating and dividing, moving and dividing at the same time. No problem whatsoever. And especially in the, I mean, if you're isolated, of course, when you divide, you don't move. When you're in an epithelium, you can stay adhered to your neighbors, divide, respread again, no problem. And I think that happens during development. I think that happens during killing. So I don't think there's any strong evidence that these are two incompatible mechanisms. Maybe Jeremy probably knows way more than I about this, because we've looked at all possible systems.
Well, the short answer is, Xavier, I don't. But but you know, I think you know, it's a sort of a bigger question. But it's not really a bigger question. I think there's a big confusion sort of out there in the world, especially people who don't think about the sort of physics of cells, which is the difference between cell movement and cell migration, because there's a lot of cell biology which is about single cells migrating, right? And I think it probably is true that, as you said, Xavier, that a single cell migrating can't move and divide at the same time because it's only got one cytoskeleton to do it with, right? But in the systems that I've looked at, you know, even when where you see, for example, intercalating cells, you still see some division, but probably as it's dividing, it's probably in the moment of dividing on that time scale, it's a passenger. And then as soon as it's finished dividing, it then carries on moving or migrating actively. Mm-hmm. So I think that's probably what it is. But I certainly think that it's on the population level, it's absolutely not mutually exclusive. Although, Xavier, you know, one of the things I was going to ask you about your analysis of the migrating cells, which is on what sort of time scale the correlation between tension and division operates. I mean, is there, you know, how long does it have to feel tension for that to affect division? Is it sort of within one cell cycle that shortens the cell cycle, or that kind of thing?
Yeah. No, so since we had so many mechanical variables and no hypothesis whatsoever, we run sort of a blind statistical analysis of knowing, well, is there anything here that is sort of standing out statistically? And the type of analysis that Marina was doing was really not looking at correlations between the duration of the cycle and the tension of the cell, but instead trying to do predictions in the sense, if I know tension at the very beginning of my cycle, which is the first I think she looked at 6 hours, 8 hours, and 4 hours. So if I know the first 4 hours, can I predict the duration in the rest of the cycle? Right? So can I make this prediction? And the answer was statistically significant, I think, after 4 hours. So in 4 hours, we can already predict what will be the duration of the cycle, which is about 24 hours. And the fluctuations in duration were pretty big. They were in between, I would say, 15 and 30. Or so, right? So it was a factor of 2. So it was quite a powerful effect, this mechanical one.
All right, let's go to Silke.
Yes, hello, Silke Henkes, Universal. And I apologize if this has been answered because I missed the first half of the talk, but have you looked at intercalations in your systems, especially in these regions where you have these extreme deformations? And if yes, what do they look like, and do they correlate with your stresses? Or do you think they actually add to it, or they follow your deformation?
You mean in the domes? In the domes,
in the domes, and also in the organoids, where you have really these extreme shape changes.
Yeah. So yeah, good. In the organoids, we don't really observe intercalations. Okay. My guess is that there is so much tension. We rarely observe divisions, but my guess is that when there's so much tension, if one cell tries to round up for division, the whole system collapses. Mm-hmm. And I would say that maybe the same thing happens for intercalation. So we haven't seen that, but maybe if we treat them if we lower tension, maybe with blebistatin, maybe if the system becomes a bit floppy, maybe we see them. It would be a very nice system to study.
It's really curious as a material then, because usual soft matter system, if you pull it beyond a certain level, it will yield. So this shows that it seems to be quite different.
Mm-hmm. Yeah. And in the organoids, I don't know how to think about intercalation because the packing is so complicated.
Yeah, okay.
Depending on the plane that you look at, you have different neighbors. So
of course,
to define a neighbor in the system is very complex because, this goes well beyond these scutoids and this type of stuff. There's neighbor exchanges all over the place. Very, very complicated packing.
Yeah, but so to the related question then, is your how spatially homogeneous is your deformation? Or is there local variability, or is it all sort of continuum level approximatively, correct?
So the organoids, you mean?
The organoids, yes. Like, could you replace this by sheet with the appropriate active mechanical stuff, or do you think there's something locally going on which wouldn't be described by that?
So I don't. Sorry, I don't understand what you're asking.
Sorry, let me try to reformulate. So if you, so how close do you think is this to a continuum system? Like, is there stuff going on there which you would expect to not be true at a continuum?
No, I think it's a completely continuous system. Okay. You can follow cell-cell junctions, so it's completely. In that sense, it's completely continuous. Except that you have some areas in which packing really deviates from what you would see in a simpler cuboidal epithelium, which we observe in the differentiated areas. But in the stem cell niche, all these stem cells, they have very complicated packing. When one detaches to divide, then it's. I wouldn't want to think about packing in the system, honestly.
Okay, fair enough.
All right, let's go to Sulin. Jean?
Okay, thank you, Andrew. This is a very beautiful talk, Xavier. Thank you so much.
Thanks.
So I do have a lot of questions. Let me ask a simple question first. So when you put these cells, epithelial cells on the hull, so you see this dome-like shape. I have done the similar experiment. I put the epithelial cells on the surface with a non-adhesive region. Okay, that's a gap. Why I didn't see the dome shape formation?
What cells were there?
MDCK.
Ah, okay.
So I suppose your experiment is an extreme case of my experiment, because in my experiment, this gap without any adhesive molecules on the surface, right?
There were no adhesive molecules, or there were some?
There's no, absolutely no.
No, in our case, there's no adhesive molecules either.
Then yes, that's why I say that the experiment setting are pretty much the same. You see this, the cell grow into the hole and then form a dome-like shape. I say the cells just close the gap smoothly, without a dome-like shape.
Okay.
I don't know why.
I don't know. I mean, if they manage, so I know one difficult thing is for them to close. But once they close, if you wait a few days, they grow. In fact, I think we discovered this by accident, because we were lazy and left a few of these samples in the incubator, and then we saw them growing like mushrooms. So, and even because in MDCK cells, if you really let them grow to confluence, you even see them without patterning. So the papers that we were discussing before with Jack, if you let them grow to over confluence, you will see them forming spontaneously, these domes. So they are very well described. So I don't know. I mean, we're happy to share protocols, but it seems that from what you're saying, what's really difficult sometimes is that these cells don't close the gaps, they do not adherent gaps. But once they close, then everyone is happy. Maybe these MDCKs are too leaky. That's another thing, that sometimes these cells are too leaky, and the junctions are leaky, so the pumping rate is not sufficiently fast to build up. So maybe that's another problem. So maybe you can try some other MDCKs. I mean, everybody has MDCKs, or you can try other cell lines. I mean, in our paper we have a list of cells that make domes. There's about 20 cells that have been described to make spontaneous domes. So maybe you can try those.
Thank you.
That, for us, more or less, it works.
Thank you. Let me add to, well, I'm next anyway. Let me add to Sulin's question. I know his paper published this year in PNAS, this MDCK cells wound healing process. And in his case, he observed that if your wound is roughly about, let's say, below 75 micron in radius or diameter perhaps, the wound would heal. Okay, so the MDCK cells would cover the area of the wound. But if the wound size is beyond 100 micron, for example, it will never heal. It will never cover the entire adhesive surface. In your case, you show that your cell could cover as large as 200 micron circular shape. Okay? So your cells must be much more, the cell-cell junctions must be stronger than his cells for some reason. But if you push it a little further, there's a limit to this. You cannot imagine that you have a wound of 2 meters, the cells can still cover that, right? So what is that limit? 200 micron, we know you can cover it. What about 1 millimeter?
Yeah.
What about
well, 200. So I didn't explain. There's a little trick here for the 200 microns, which is to add a little bit of matrix in the non-adherent gap. So instead of making something that is binary with a lot of matrix and no matrix, we do a lot of matrix and a slightly amount of matrix. Otherwise, we do not close the wound. And that was the clever thing that Arianna found out. So that's important. That's when, but it's, of course, you have to add sufficient matrix so that cells cover, but low enough so that they can then build a dome. So it's a very delicate balance that Arianna found a way to work. And then, so yeah, on a surface there might be a limit, no, that you can invade. And I don't know what this limit is, and what sets it. I guess the surface tension that you have in the periphery that must overcome. I mean, must drive cells overcome all this friction of cells to move in, right? So I don't know. I guess it's that's the mechanical balance, right? Because that's what you have. You have a proliferation rate, but at the end you have a ring that is dragging cells, and there's a friction and a viscosity there that you need to overcome. So I mean,
right, I guess that's the difference between your experiment and Sulin's experiment. In other words, his surface, his wound is non-adhesive at all. Yours is slightly adhesive.
Yeah.
That's one thing.
Yeah. By the way, I'm Jimmy Xia from Nanyang Technological University in Singapore. And Xavi, fascinating talk, very interesting.
Thank you. Thank you.
Thank you so much. I have a follow-up question on this wound healing process. So you form a dome. Since the dome has a curvature, the pressure inside the dome must be different from the pressure outside the dome. And that pressure is balanced by surface tension of the dome, which you showed. And it's very easy to imagine that as the dome grows, the curvature becomes larger, or the radius of curvature becomes smaller. So in order to balance the pressure difference, you need higher tension in the dome. Okay? And you actually showed that. You showed this sort of a linear increase of the tension in the domed membrane, in the domed cell sheet, until you reach a plateau. At that point, it stopped increasing. Wouldn't that be the intrinsic maximum cell-cell junction force that you are measuring?
No, so, but once you reach that point, I mean, you still deform, the system still deforms, but tension no longer increases. Right, so you're stretching at constant tension in that case.
So I guess that the cell has a tensional homeostasis. You want to maintain certain level of tension to survive. Is that something that you're thinking, Xavier?
Yeah, go ahead, Sulin. Yeah.
Well, I'm asking Xavier.
I think there are two effects. One is that as the system stretches, there's one geometric effect, that as the system stretches, there is an increase in tension. So geometry plays a role, and so this is a three-dimensional epithelium, and therefore there's the system is the two apical surfaces get closer, and then so so there is a relationship between apical and basal surface tensions and lateral tensions. Right, so these are not the same. So as you stretch the system, the apical and basal areas increase, and the lateral shrink. So there is a shift in the relative contribution of these tensions, and this is in fact a geometric effect that causes an increase in the tension of the monolayer, simply because you go from a system in which the dominant contribution might be the lateral tension to one in which the dominant contribution is the apical tension. And this is just because changes of area, right? So that's so when you write the energy of the system, that's what happens, right? So that's one thing that would make the system tend to increase tension with strain. But then there's another effect, which is the softening effect that we were mentioning before, which is that some cells at some point they start becoming super stretched, and then for these cells soften. So you have this combination of two effects. One in which the system wants to increase stiffness, but another one in which some of the cells start popping up, and then that the system softens because of that. And I think that the plateau that we reach is because of that. So I don't know if Sohan Kher is here. He developed a model that explains this. So if in any moment, Sohan, you want to jump in here, because this is your baby. So I'm just trying to explain your model.
No, I think you're doing a fantastic job explaining it. Yeah, I think the plateau that we get is because we have two different possible phases because of softening, one which is at the low strain and one which is at the high strain. And as we stretch the monolayer, the plateau corresponds to having mixture of super stretched cells and barely stretched cells. And we will stop the plateau as soon as all the cells have gone super stretched, and then we'll start reaching this hardening regime where all cells are responding in their post-softening response, driven by intermediate filaments or squeezing of the nucleus or running out of the membrane material to replenish.
Mm-hmm.
So I was wondering whether or not the activeness of the cell play a role in this process. So it seems to me that so Han, that your answer, your attribute all this phenomena to a passive response of the cell. Is that my understanding is right? Where is the activeness comes from?
So
do you think that way?
So the softening is actually it's driven by an active process, because how we understand it is that as a cell stretches more and more, there is more total surface area for the cortical material to cover. And at some point, as Xavier explained, the amount of actin monomers that the cell has are not sufficient to maintain this cortical thickness. And the cortical thickness is not the same as it was initially maintained at some value driven by the turnover of the actin material. And as a result of this, as we stretch more and more, the amount of cortical density decreases at some point at very high stretches. And this would lead to a softening response. And since what drives the cortical tension is an active process, I think the origin of this softening is also active in that sense.
Right. So we reason in terms of actin polymerization, depolymerization here, but at the end, what sustains tension is myosin pulling on actin there, right? So and this leads to, I mean, our model is extremely simple, right? But one can think that if you start stretching actin, and whether myosin becomes more or less trapped in that network. No? So we have on purpose ignored or strongly simplified the actin cortex, which is a very complex material, right? But when the cell stretches, what does the actin cortex do? I think this is a super important question that we deliberately ignored in our paper with some very simple assumptions. But we need structural data to know how pore size changes, whether myosin motors can still generate force when you stretch. So it's a very complicated material, right?
Fascinating, but even engineered materials, at least some engineered materials, polymers, would have such a behavior. When you most polymers, when you load it very slowly, very little, you have an elastic range. Then when you load it beyond a certain range, all of a sudden you have this polymer chain sliding behavior that undergoes very large strain until you hit the point when the polymer chains more or less aligned with the loading direction. Then it goes up, the resistance goes up tremendously.
Exactly. Yeah. So exactly, and that's exactly, and incidentally, that's, for example, also what intermediate filaments do. So if you take a single intermediate filament and you stretch it, that's exactly what you have. And first you start stretching bonds, but then you have a transition where you have, it's not so much a sliding, but you also have transitions between alpha helix and beta sheets and stuff like that. But eventually everyone stretch, and then the stiffness of the material. So I think this, so this increase in tension followed by a plateau, followed by another increase, this happens in engineering, and it happens in living systems at different scales, which I think is really interesting that it is so conserved.
Beautiful, beautiful. Thank you so much, Sai.
Kari, I have another question. I have a lot of questions, but I'm going to ask another question. Please allow me. Okay, I'm going to communicate with you later on. So when you culture the cell and the cell differentiate and proliferate, and you measure the traction force or monolayer stress using traction force microscopy and monolayer stress microscopy. So you say that generation 1, generation 2, they have different traction force? Do you see any plasticity there? We'll all talk about elasticity. I would like to say, I'm wondering if there are any plasticity there, meaning that generation 1, generation 2, when you expose them to different phenomena, or when these cells experience different loading history, did you say they experience or they exhibit any plasticity behavior?
So you mean mechanical plasticity in the sense that when we stretch them and we bring them back, are they different or?
No. I'm talking about another experiment that you have done. Say, remember that your cultured cells and the cell will proliferate at the same time. You have a generation 1, generation 2, right? And you measure the traction force, right? So the first question, simple question is that when you measure the traction force, while your cell proliferate, the traction force, the magnitude are the same for the generation 1 and generation 2?
I would guess so, as long as they duplicate their material. So I would say that that's what's the case. Another thing is that you can have some sort of history in the sense that, for example, cells are continuously not making extracellular matrix. We know that the amount of matrix that they make is important for the traction that they generate. So I think that one way in which there is some plasticity or history to the systems is by the amount of ECM that they are able to generate and to accumulate. And there was a nice paper by Benoit Ledoux on this related to cell migration. Otherwise, I think that.
I'm just thinking that aging is a natural plasticity. It's this plasticity. I'm just thinking that whether or not, if you can tell whether or not cell undergo plasticity from the traction force generation. It seems that to me that your traction force pretty much remain the same. I don't know.
They do. Yeah, I don't think it's a good readout for generation. I don't think, no.
Okay. Yeah, I let ask people. I ask people to ask their questions. Very, very fascinating talk. I'm going to talk to you later on.
Absolutely.
Okay.
Let's go to Pierre.
Thank you. I'm Pierre Haas. I'm at the Max Planck Institute for the Physics of Complex Systems and of Molecular Cell Biology and Genetics in Dresden. I was actually wondering, how do you think about the leakage or the fracture mechanics, for example, in luminogenesis, during organ formations, some cells actively rearrange their tight junctions to do something. Do you expect to see this, or do you expect the leakage to be an actual mechanical failure?
I don't know. What do you expect?
I have no idea. I actually don't know what the mechanical. I suppose you could somehow estimate the stresses, but I don't know numbers for the forces that tight junctions can sustain. Actually.
Right. Yeah, as I said, all these fracture mechanics problems are extremely hard to control or to even visualize, right? Because one of the problems in physiology, in epithelial physiology, right? One of the main problems in epithelial physiology is when an epithelium becomes leaky, right? And when that happens, people who study this in the lungs, for example, the gaps that form are on the nanoscale, right? So they can tell about them by looking at the permeability of really, really small particles. Like as soon as they go to, I don't know the numbers, but as you go to 30 nanometers, they are no longer leaky, right? So you have to really go to is so small, this is so nanoscale that probably, I mean, you're probably right in the sense that modeling predictions would be very helpful here. I mean, here's where modeling would be useful in saying, well, this cannot happen because we know that junctions are this, and that. So this cannot explain the flows that we observe. What I'm afraid of is that the flows that we observe are so weak that there can be many explanations to them.
Just a comment to Pierre's question. Fracture mechanics at that small length scale may not apply.
Yes.
Mm-hmm.
Actually studied that years ago.
Mm-hmm. So at that length scale, you mean nanoscale, or I mean in the hundreds of nanometers, right? That's
or below that. Yeah.
Yeah. No, we do observe sometimes these sort of bubbles in between cells, right? They are microns. So in this case, this is some sort of fracture. I mean, this is fracture mechanics, right? But yeah, I agree. So how do you call these small pores? How do you interpret them? Whether they will grow or they will not grow? That is probably a problem for the experts in fracture mechanics, which I'm certainly not one of them.
Jimmy, I have a question quickly in response to your comment. Do you mean the continuum description of fracture mechanics will not apply, or we can think of these as fracture events?
The continuum mechanics based fracture mechanics. In other words, fracture mechanics is based on the theory that you have a crack, you have certain type of singularity field at the crack tip. If the length scale is too small, that particular description fails.
But it's obvious that it will not work because the system is just not a simple mechanics system. You have to, there is flow, there are electric field, there are, the cortex, you have all these things have to be taken and, together. And so there's just no way you can apply the knowledge from some other field.
Right.
You have to construct that.
Right.
We have to do it. I mean, or you have to do it. You're young enough to do it.
Exactly. So for the cell system, if you want, this is a highly irreversible process. Energy comes in, energy comes out. Whereas in classical fracture mechanics, you have the energy release rate. I mean, this fracture crack propagation. That's surface energy gain, you have the strain energy release, and that's the only mechanism in classical continuum mechanics. Whereas in this highly irreversible process, it's a lot of energy sources comes in, comes out. So
of course.
So maybe you can construct a microscopic model in a vertex model or something, a fracture in a model like that, and then take into account, I don't know, fluid structure interaction or some other things where you don't completely neglect. I mean, it's going to be very hard, but maybe.
Well, yeah, probably it will be a mesoscopic model.
Yeah.
You have to go to small scales, but maybe not to the smallest.
Okay.
And you have to do it.
But like a vertex model would ignore, like
ignores
Philopodia.
Too many things. No,
I mean, whenever you have these voids, I mean, you should expect that you'll have some sort of, activity of the cells to try and reinstantiate their neighbors and reassemble their junctions with, naively.
Yeah, no, vertex is good for some scales, but I think for this kind, I don't think it would work. So I had a few very naive questions.
Go ahead, Jack. You've been patiently waiting. So yeah.
So the first one, I mean, I love this flat villi. I mean, this flat villi experiment is just gorgeous.
Thank you.
So a very simple question. Did you look at the total integrated normal force and total integrated tangential forces?
No. It would. I mean, you mean at the total at the periphery of the.
If you integrate, let's speak of the normal force that you measure beautifully. If you integrate that force over the whole area,
does
it add up to 0 or not?
it better adapt to 0.
Well, no, not necessarily, because it's an epithelium. And if it did not add up to 0, it would tell me that the pumping activity is important. I mean, that's some importance. It would give me a hint on the whether or not I can forget about the pumping activity.
But okay, but how pumping balances attraction?
And then it depends on the permeability of the gel the system is sitting on, and so on. I mean, it's not the outcome is not trivial.
Yeah. No, the gel is not permeable. The gel is not permeable. If the gel is permeable, then it's very you don't form these domes.
Okay.
So we use PDMS to ensure that there's no leakage. At least that's one variable that we can get rid of is that.
And then it tells me that you can generate a lift force, either positive or negative. And it would be nice to know at least how big it is, the sign and so on. That's one thing. The other question is, did you try to measure the electric potential difference across the epithelial layer and see whether it changes from region to regions?
No, but I'm super interested in this topic.
Okay, I would love to. Okay.
We haven't done it, but I think we should really incorporate this to our descriptions.
Okay, you think you can do it?
I'll talk to a few people. So that Daniel Cohen was here before, so there's a few people who can do this.
Oh, that would be really great. And maybe last, you said that you've done an experiment in which you pump water into the cells. Can you tell us a little bit more? If we are allowed to.
Yeah. Of course. Yeah.
And can you maintain them over very long time, like days and weeks?
Yeah.
Which would be really.
Yes. So we're working on a microfluidic device in which we grow these domes, but they are sitting on top of a substrate that has gaps on it, and therefore through this microfluidic device we can inject pressure from the bottom of the dome, and therefore we can inflate at any rate we want to. We can sustain it for as long as we want to.
And can you change the sign as well?
No.
Okay.
Not now, but we haven't thought about it. Not now in the way it is made, but it should be doable. I need to ask. We have a fantastic PhD student working this out, so hopefully we'll soon be able to start reproducible data.
I think that would.
But yes, we
can do
that
it for a long time.
Yeah. So we can sustain this pressure difference for a long time.
Okay, that's fantastic. Yeah.
Yeah.
Very nice. Well, thank you and congratulations.
Thanks, Jack.
Thank you. Let's go to Teng.
This is Teng Zhang from Syracuse University. Thanks, Xavier, for the excellent talk. I have a question about the modules of different tissue layers. I think your system can measure like the active tensine, modulus surface tension of different tissue layers. So I was wondering whether did you do this kind of experiment, especially one example is for the skin. We know that there is a multi-layer like epidermis and dermis layer. We did some study on the wrinkle on the torus to mimic this like a phasor pattern, a scale pattern in the embryo development of like a chicken and the crocodile. So we found that a purely mechanical system, like you mentioned, is a differential growth, can trigger a similar pattern in the real biology. But we have very small modulus ratio between epidermis and dermis, maybe 5 or 10 times. when I search literature, most data are based on the mature skin. So that ratio can be 1,000. So I didn't find a reasonable modulus during this embryo development, very early skin stage. Thank you.
So what you so I guess your question is whether you can measure the surface tension of different tissues, and whether you can find these large differences that you need for I mean one so you talked about the skin, no? So one problem is that the skin is stratified, right? So there's multiple layers of skin which will completely change. I mean, we could not probe skin in our device currently because skin is not pumping, to my knowledge. Maybe where there are blisters in the skin, so, but I don't think that comes from spontaneous pumping. But in the other case, it's much stiffer than what we could probe in our system. But with the device that we were discussing with Jack, in which if we can't really control the pumping, then we really can turn this into a rheometer where you can plug in any tissue, and then you can actually measure very locally the properties of the surface tension mostly, but essentially the stress strain curves in the system for extreme regimes where you go up to 300% or whatever. Yeah.
Okay, I see. Yeah, thank you.
All right, and then let's go to Yaling, the last hand.
Right, it's an honor to be the last. I actually jumped back. I've been watching for a while and have many. So I'm from Lehigh University. An excellent talk, Xavier. I have a question about you. One of your slides you were mentioning about the interaction between the fibroblast and the tumor, right? So that's obviously a popular approach. People usually co-culture tumor with some other cells to form organoid. And also, tumor itself is very metallicity. They will migrate as well. I mean, from your movie, are you suggesting that the fibroblast will help the migration or excavation of tumor and make it move even faster, or what did that help?
Yeah, so that was really surprising to us. I mean, that's one of the most strange and unexpected results. But the answer to this question is not black or white. So we show that there are mechanisms in which fibroblasts, and Eric Sahai has published a lot on this, where there are mechanisms in which fibroblasts help cancer cells progress. And this is somehow puzzling because you would expect that cells that are not tumor cells, what they do is they prevent progression of the tumor, right? And so now what we do is that the signals that tumors generate can sort of hijack the functions of these healthy cells and then help them progress. So in this case, having fibroblasts is bad, and there's many reasons why it is bad. But another thing that fibroblasts do is that they envelop the tumor. So in some tumors, you see, and that's again back to Daniela's data, where you see really that there's fibroblasts surrounding the tumor, and that prevents their growth, right? So it sort of compresses them. So there's this double phase of these fibroblasts that sometimes they help, sometimes they don't, and there is a discussion in the field. There's an important discussion in the field on how you target fibroblasts. I mean, are they good? Are they bad? And fibroblasts, they also make a lot of matrix, so they make the tissue much more stiff, and they're very contractile, so they completely mess up the microenvironment of the tumor. And so I think, so trying to understand whether they are good or bad, no? You know how medical doctors are sometimes. Are they good? Are they bad? Because they need an answer, because they need to act, right? And there's no clear consensus there.
Thanks. Sounds like interesting.
In fact, we have a paper in the archive, in the bioarchive with Daniela Bienevich, on this exact topic. So on the role of mechanics of these fibroblasts in the progression of the tumor. So if you want to take a look, that's in the bioarchive.
Great. Thank you. We'll definitely check it out.
Thanks.
All right, great. So I see no more raised hands. And given that we have been on for almost 3 hours, I think this is a good time to stop unless there are any remaining burning questions. So I want to thank Xavi for giving a wonderful presentation, and also to all the panelists for this really wonderful discussion. And then I'm going to give the word back to Jimmy, who may want to announce the next webinar.
Right. Thank you. Thank you very much. So our next webinar is going to be given by Andrej Kosmrlj of Oxford University. And the discussion leader for that particular webinar would be Krishna Karipati of University of Michigan. So it's going to be on Wednesday, October the 13th. And again, starting at 10 a.m. and informal interview at 9:30 a.m. Thank you again, all of you. We always have some diehard, and we have excellent discussion today. And everything is recorded, so people who are interested in this can always go back to watch the recording, watch the video. Teng, do you have anything that you want to mention?
Thank you, Jimmy. Thank you, Ravi and Andrej, for this wonderful EML webinar and the interview before the webinar and the in-depth discussion afterward. Just to let you know that we do the live streaming in WeChat as well. So far, there were more than 7,000 people watched today's EML webinar. So congratulations for the wonderful event brought to us.
Great.
And everyone who participated in the discussion as well.
Okay, thank you all again. You have for some people, good night, or others, a good rest of the day. Thank you.
Thank you.
Thank you.
Thank
Bye-bye.
you.
Bye.
Thank you very much.